Good morning, everyone. It's a great pleasure to see all you here, and it's my honor to open the first edition of the PhD Food Arena, the PhD program in food science. Briefly, what is the meaning of this meeting? Is somehow rely on the experience of the Gordon Conference. I don't know if you are aware about it. Gordon Conference are very specific conference that are usually held in California, so nice place, where they select one topic and they invite no more than 40 scientists from all around the world. They close all the scientists in an hotel room, an hotel area where they can show their research products. In which way? Something that's never been published before. So the fundamental approach of the Gordon Conference is that you can bring out any kind of data that probably you will never publish, any kind of hypothesis, and have a discussion, open, free discussion with all the other scientists in the area. And everything stays there. Nothing is published. So it's really a kind of meeting where everyone can say whatever he wants. And this is the idea of this PhD Food Science Arena, a place where every one of you can present your data, your hypothesis, what you have done, what you will done, on the basis of the area, the topic that you are tackling. And also, is more than welcome that everyone can ask questions, provide advices, any kind of. Because, you know, the strength of this PhD program is really food. And food is somehow a topic that can be faced from different point of view. And each one of you has a different expertise that can be extremely valuable for everyone. So this is the idea. Don't be shy. Make question, make comment, any kind of. We are recording, but, you know, don't be shy. So briefly, I don't want to spend more time because it's time that we start. So I will invite here the magnificent human being, director, Professor Dino Mastrocola.
Good morning, just few words, but in Italian, I'm sorry. No, veramente due parole di saluto e sono particolarmente contento di essere qui, tenuto conto che questo è anche il mio dottorato di ricerca, quindi, no. Volevo anche complimentarmi per questa iniziativa, perché come diceva Mauro è un momento di confronto e per ognuno di voi, indipendentemente dall'anno in cui vi trovate, insomma è un momento importante di confronto, ma proprio anche per focalizzare, mettere il punto sulla ricerca in atto, però penso che sia anche utile per gli altri poi ascoltare in modo più dettagliato quello che si fa all'interno del dottorato. Quindi non posso che augurarvi buon lavoro e quindi che sia una giornata proficua, anche in vista poi del workshop nazionale, che questo penso che sia uno dei pochi dottorati che da diversi anni si confronta proprio a livello nazionale, dando ad ognuno di voi poi la possibilità di presentare i propri risultati. Mi scuso ancora per l'italiano, ma sono stato colto di sorpresa. I'm sorry. Ciao.
Grazie Dino. I'm invited on the floor Professor Danese, Director of the Department of Food Bioscienze.
So good morning to everybody. So actually, it's a great pleasure for me for being here. And, you know, starting this, I would say, new, fashionable way to exchange science. So, of course, as Mauro was telling you before, it is a model already applied by the Gordon conferences that are actually also held in Italy, in Tuscany, in Barga, that I was being there in 2011. And it was a great pleasure to participate to this kind of conferences because, as Mauro told, it's a free way to exchange science among different disciplines, among different topics, expertises, and so I guess that this is the best way to do science. And, as a young investigators, you are the best example we can have here in Teramo. And I guess that this model, we were discussing about this model of presentation during the meeting of the president of the coordinators of the different bachelor and master degree courses, and we were inviting also the PhD coordinators during this meeting because we were starting with a new phase, with a new model of sharing science among the bachelor, the master degree courses, and the doctoral program. So we were starting to see what kind of courses you have to follow the test to be different from those that are present in the different bachelor and master degree courses. And also these seminars that and the information that are coming out from this kind of meeting are valuable also for our students, so to share the information among different students in our department. So I wish you a very pleasant and nice discussions and also fruitful from the scientific point of view. And I see in your eyes this very enthusiastic approach, so I guess that will be a very nice day. So thank you again for being here.
Thank you very much. Okay, just a few words. Of course, this is the first edition, so we will really welcome your feedback about it, because the idea is for the next edition also inviting stakeholders that can be interested in your research, that can support PhD fellowship and so on. So in our mind, it will be something a little bit more organized and open to outside. So really, any kind of feedback you have it, meet me, send an email, whatever. Feel free to express yourself. So I will start with the presentation. And the first one is Dr. Buccioni. Title is Enhancement of Antimicrobial Bioactive and Technological Properties of Lupin and its Byproducts.
Okay, so good morning, ladies and gentlemen, professors and colleagues. It's a pleasure to be here today and an honor to be the first PhD student to participate. Also a responsibility to be the first PhD students to start this event. Today I will briefly talk about my PhD program and project that is about, as Professor said, the enhancement of antimicrobial, bioactive and technological properties of lupin and byproducts. So, as you can understand, the main character of the project is lupin that has both a good side and bad side. For the good side, I will spend just a few words on the activities I did on my first year that were about the assessment of an antimicrobial activity of lupin. We tested different lupin extracts and different protein isolates from lupin, but low or no activity was identified, so we switched our focus research on something else. So only some data showed in the slides. Okay, so with lupin, we briefly talked about the good side, but there are two bad sides that are represented by Dipartimento toxica. There is a phytopathogenic fungus that has lupinous species plants as major hosts. The problem with this mold is that it produces phomopsin A, that is a mycotoxin that causes a disease called lupinosis. Lupinosis is mainly for animals, let's say, and can affect kidney and liver. In recent years, since the consumption of lupin has increased, EFSA, let's say, produced some opinions in which this authority needs for a better identification and quantification of phomopsin A in lupin and lupin-derived foods, and the New Zealand authority, there is the only authority that produced some regulation about that, fixed a limit of 5 ppm for phomopsin A in lupin and lupin-derived products. The other problem with lupin is the presence of quinolizidine alkaloids, that mainly are the four represented in the slide, that are a class of chemical compounds produced by the plant during its growth as a defensive mechanism against pathogens or insects or other problems it can go. So these quinolizidine alkaloids can act when ingested by humans as acetylcholine receptor agonists, and so decrease the coronary flow, the contraction amplitude, and the heart rate, so they can be potentially harmful for humans. So my activities in the second year were about the reduction of both phomopsin A and alkaloids. Okay, so modern problem require modern solutions, so we wanted to use modern technologies to reduce these two issues, let's say. The first one was ozone. The first we characterized the mold, okay, on different media, and wanted to analyze the growth and the reproduction of the mold. We observed that the best media is lupin, of course, and on other media the growth was good, but the mold did not produce spores. Anyway, also on this substrate, the mold was not able to produce enough spores to inoculate lupin for the further analysis, so we used the fungal mycelia to go on with the analysis. Okay, as I was saying, the first technology we wanted to test was ozone, and we tested it in aqueous solution until now. And according to literature, the best concentration was 7 ppm for 4 hours. We had a good reduction, let's say not great, but good reduction of almost 20%. That was good in our opinion. But the great results were about alkaloids, because we had a great reduction of all the alkaloids tested that was also significant statistically. The second technology was the pulsed light. We evaluated the effect of the pulsed light employed at different parameters on lupin samples, and we had some reduction in our samples. The problem was that the reduction, we were not able to see always the relationship between the reduction and the intensity of the pulsed light treatment. This was true not only for phomopsin A, but also for all the alkaloids. So the objective in the third and last year of my PhD is to go on with the analysis on ozone and pulsed light by also optimizing the method for pulsed light treatments. The second and the other objective we had was, since the lupin is being consumed more and more in last years, was to produce a lupin-based snack. Of course, I will not show the formulation because they are sensitive data for the company, but I will show some results we had on the snack. So first, we had to produce different recipes for the snack and to identify the best one. We used as for the evaluation some sensory evaluations, and from a visual and olfactory assessment we had not really clear which was the best sample, but from a flavor point of view, we observed a really good trend for the sample 25. That was the highest in sweetness, had very low bitter flavor and burnt flavor, and was the highest in crunchiness, chewiness, and crispness. This trend was confirmed by the final evaluation on the panelist acceptance of the snacks that confirmed the sample 25 as the best one. Once we evaluated the snack, we wanted to optimize the cooking of the snack, and we evaluated different time temperature combination, observing that in some cases the snacks were burned at too high temperature. So we choose to work with the combination 160 degrees for 10 minutes in a professional oven. After the production of, let's say, the best snacks in the samples we had, we wanted to add salt to the formulation at the minimum concentration possible and add a dry seed coat to increase the fiber content of the snack. So we performed a central composite design with 2 factors at 3 levels, and we identified as the best percentages the 2% of dry seed coat and 0.67% of salt. Okay, finally, so in the objective for the next year that already began, but okay, there's for sure the optimization of the methods for the pulsed light and the evaluation of other parameters for the ozone and pulsed light effect on both phomopsins and alkaloids. Concerning instead the production of the snack, we wanted to use different flavors to enhance the properties, the flavor properties of the snack, and to test, to perform some accelerated shelf life tests in flow packaging and modified atmosphere. So here I put some other side activities of the project with more activities more related to the project that are yet to come. For example, the publication of a review article on lupin and some oral communication I had on national and international congresses. Thanks everyone for the attention.
Thanks, Francesco. Any question?
Yes.
First of all, thank you for your presentation. You presented very interesting results. I have just a curiosity if you had the chance, in relation to the ozone treatment, also to evaluate during time the decay of this reactive oxygen species.
We had not till now, but for sure is something that is mandatory to do before any other analysis, because we cannot suppose that any residue of ozone is in lupin. And we need to evaluate the duration of the effect of ozone on our samples. So.
Just another thing, because maybe I lost this information. Did you perform the treatment on model systems? I mean, with lupin or just in model systems?
No, we inoculated lupin, okay, with the mold, okay, in order to produce a high quantity of mycotoxin, and we treated the lupins, the inoculated lupins. Okay.
Any other question?
Thank you for your presentation.
You're welcome.
I would like to ask if you are going to evaluate if the treatments with the pulsed light or ozone can be an effect on the components of lupin from the, not only from the organoleptic point of view, but of the nutrition components, if can be a negative effect of such of these components.
Okay, suppose you mean like proteins or
Yes,
lipids,
or,
okay.
vitamins. I don't know really the
Okay.
But
Yes,
guess that this treatment oxidative can be an influence on the composition.
Yes, I really would like to. Is not actually part of the project, but is a really interesting analysis. So if we had time or we are going to evaluate that for sure. Thank you.
Any other? One actually is more or less the same, because, you know, what it was saying is correct, because usually when we deal with food, you know, it's a complex word. So maybe you solve a problem, but you raise another one. And the ozone treatment, that is free radical inducing protocol, it clearly has a clear effect on alkaloids, but it will be useful checking just with a simple antioxidant properties testing, you know, because maybe you are declining all the other functional products that are in the lupin, maybe not, because maybe it's specific, but I will run a test on this.
Yes, if I can only joke, is of course a kind of balance between
right.
the issue you want to solve and
right.
effect.
That's why you need to have that data.
Yes.
Because maybe, you know, it's like when you put something like you know, lettuce under modified atmosphere packaging, in a few days all the antioxidants disappear. So you have these products that can last forever, but has nothing inside. So it's a matter of deciding the balance between the intensity of the ozone, or maybe, you know, ozone is not so strong, but, you know, it might have an effect. So, I mean, this is a part that I think you have to consider. Thank you. Okay, any other? Okay, thank you very much.
Thank you.
Did you say that, if I'm not wrong, you said that you had a liquid system with the
With the
ozone
ozone?
treatment?
Yes, we dispersed the ozone in water. So, and okay, we put lupins inside this chamber.
Did you hypothesize to work with gas, just with a gas activated? So
For instance?
I mean, you can produce ozone and just use ozone, have air with a high concentration of ozone inside. So
Just
without
with the air?
the use of water.
Yes, we are actually waiting next week the instrument to measure also air ozone concentration in air. So we will work with the same chamber but without water.
Okay. Because maybe you could also increase the effectiveness
Yes.
of the treatment in this way.
Okay.
Thank you very much.
Okay.
We move to the next speaker, Fiori. That will talk about development of flexible sensor based on nanomaterials obtained using natural compounds on substrates with minimal environmental input.
Can you see my presentation?
Yes, we can. Yes.
Okay, so good morning everyone. I am Selene Fiori, a PhD student in food science in the group of analytical chemistry.
La puoi mettere in modalità presentazione così?
Sì.
Okay.
Okay, perfect. So my doctoral project is about the determination of a compound of importance in the field of food quality and safety, thanks to the use of electrochemical sensor. The development of a sensor is based on both the sensing material and the substrate. As for the sensing material, the most commonly used are carbon nanomaterials such as graphene, carbon nanotubes, or carbon black, whose properties can be implemented thanks to the integration with other nanomaterials in particular metal nanoparticle, transition metal bichalcogenide, and metal oxide. The substrate can be of different materials, such as plastic or, in recent years, paper and recycled paper. In literature to date, exists a lot of electrochemical sensors proposed for the food analysis, and the trend is to find new sustainable and renewable material and production techniques that are solvent-free, easy to use, and low cost. So in this perspective, I will show some of the work that I've done in the last years, in which the developed sensor is applied for food analysis. The first example is about food quality, in particular the valorization of food waste and byproduct rich in bioactive compound. In this case, the concept used is the one of circular economy, in which a byproduct can be used as the starting material. In particular, plant-based waste derived from paper industry are used for the production of a paper-based biochar sensor. Biochar is a carbonaceous material that can be produced with pyrolysis at controlled temperature and atmosphere from carbon-rich biomasses and agroindustrial waste. Biochar is subjected to an ultrasound-assisted liquid phase exfoliation in water in the presence of a stabilizing agent in order to obtain a highly conductive nanofibrillate biochar. The biochar dispersion was then filtered in a cellulosic membrane, and using a laser plotter, the working electrodes of biochar were obtained. Then on the substrate, that is paper fully recycled from cellulose, the contents were printed using a stencil mask. And to complete the sensor, the working electrode were integrated onto the substrate. So the application is in the determination of polyphenol compound that can be found in food waste, in particular cocoa husk, coffee husk, exhausted coffee pod, and also food supplement that can be produced from food byproducts that are olive leaves and artichoke leaves. The quantification was performed with the standard addition method using the electrochemical technique of differential pulse voltammetry. So in this case, we obtained quantitative recordings proving that the sensor can be used for the valorization of food waste. Another topic is the analysis of food safety and also of food grade surfaces. In this case, we proposed a sensor based on reduced graphene oxide and cereal nanoparticles used for monitoring of hydrogen peroxide during the disinfection treatment from listeria from food-grade surfaces. The production of the sensor starts with the contact printing of the substrate, that is nitrocellulose, and then the preparation of the sensing material, that is reduced graphene oxide and cereal nanoparticles obtained with a CO2 laser plotter that can induce both the reduction of graphene oxide and the formation of cereal nanoparticles. Then the sensor, also in this case were completed, integrated the working and the counter electrode onto the substrate by pressure. So why we have chosen nanocereal? Because it shows catalytic properties towards hydrogen peroxide, thanks to its ability to switch between two oxidation states. The measures were performed in a box in the presence of two different strains of listeria and using a nebulizer to apply the hydrogen peroxide. The monitoring of hydrogen peroxide concentration was performed in real time using chronic amperometry. So by varying the initial concentration of hydrogen peroxide and the time of application, we can relate the area under the amperometric course with the effectiveness or non-effectiveness of the treatment. Then the last section is about the monitoring of food safety, in this case with the analysis of a pesticide, the carbaryl, that can be often found in grain. The device proposed is a paper three-dimensional pop-up device integrated with a sensor in reduced graphene oxide for the determination of the carbonyl. The device was obtained using a laser plotter to cut all the components and a wax printer to obtain the hydrophobic area. Then the laser was also used to reduce graphene oxide, and the sensor was fabricated in a similar way to what first reported. For the measure, the device was opened in a three-dimensional configuration, and the sensor was placed in a holder. Then, onto the sampling windows, we have consecutive deposition of the sample extract in order to obtain a concentration of the carbonyl thanks to the evaporation of the solvent. Then we add the sodium hydroxide to hydrolyze the carbonyl into one naphthol that is the electroactive form. We close the device in the two-dimensional configuration, add the buffer to neutralize the pH, and then measure with the differential pulse voltammetry. We analyzed 5 different types of grains with the standard addition method, working at 3 different levels of concentration. That are the legal limit of carbonyl, called minimal residual level, half the limit, and 1 time and half this limit, in order to correlate the quantification performed by your sensor with the safety of the sample. So now what I'm doing is an abroad period in the University of Regensburg, working on the development of a laser carbon nanofiber electrode integrated with nickel nanoparticles for the non-enzymatic detection of glucose in butyresol. And then I will also do a period in Sense4Med, that is a company in Rome. This is the summary of all the work that I've done to date, and the conference and workshop I attended with poster and oral presentation. So that's all, and thank you for your attention.
Thank you very much. Any question? Okay, I have just a curiosity. Do you think that can be commercially applied in terms of costing? The assessment or the carbonyl or polyphenol of H2O2?
Yes, it can be potentially applied also commercially. Even the costs are very, very low. We are talking about less than €1 per device. So yeah, it's possible to eventually have commercial product.
Thanks. Okay, thank you very much. We move to the other speaker, Tata Shore. Innovative formulation and process approaches for new plant-based ingredients with enhanced techno-functionalities.
So, good morning, everyone. As the title states, my project is based on the development of plant-based ingredient. But why we have to produce plant-based ingredient? As a short background, because consumer awareness are more aware about the impact of the diet on their health. So they start to read carefully labels, and they ask food industry to replace the use of synthetic additives with natural counterparts like bioactive compounds, generally extracted from plants and herb with the techno-functional properties. But to extract these bioactive compounds, a tailored and optimized extraction process is needed. And today we are looking more and more to green extraction technologies. But there is a problem, that these bioactive compounds, if compared with synthetic additives, are sensitive to environmental stress, like exposure to high temperature, oxygen, and light, every factors that occur during processing, storage, and transport of this ingredient. In addition, some of these molecules have a very low water solubility and very strong astringent and bitter taste. So, what could be the solution? To use microencapsulation. What is microencapsulation? Is a technique that enables to enclose these bioactive compounds inside a structure in the microscale in order to try to overcome all the drawbacks, so to protect the bioactive compounds, in addition to modify the physical characteristics of the initial extract that generally is not easy to handle. We can also control the release of these bioactive compounds and mask the unwanted flavors. To make this, different kind of encapsulation technologies can be explored based on physical, chemical, or physicochemical method. And to perform the encapsulation, we need some material that give the structure to the microcapsule. And we are speaking about, of course, food-grade compounds, but of different nature, protein, lipids, carbohydrates, and generally they are also mixed together. So to obtain the microcapsule, our optimization between the right encapsulation technologies with the right encapsulating agent should be chosen. So the plant from which we decided to extract our bioactive compounds is the hop, and in particular the female inflorescent, the cone, which is full of this yellow powder which have different kind of bioactive compounds. Here we have an overview of all the bioactive compounds present in the hops, like volatile oil, phenolic compounds, among which hop possess a specific class of compounds, prenyl flavonoid, and among which xanthomumol is one of the most characteristic and abundant, and bitter acid, alpha and beta acids. All these compounds possess techno-functional properties. Functional from a nutritional point of view, but also a techno-functional property. What does it mean? That, for example, bitter acid exert foam stability activity. So, what is the project objective is to explore different kind of extract and encapsulation process to obtain this hop-based ingredient to use possibly in food formulation to obtain products with improved qualitative and sensorial properties. To reach the objective, my first activity is the study of the chemical composition and the technological functionality of different food-grade hop extract, and the second one is the evaluation of different encapsulation technologies to produce the hop-based ingredient. This year, I focused on the development of a HPLC DAD method able to simultaneously identify and quantify with 31 bioactive compounds, in some of them specific in hop, and this method helped me then to focus on the single compounds of the hop, both in the extract and in the obtained powders. So, starting from the first activities, these are the two kinds of extract that I studied. A hydroethanolic hop extract obtained by ultrasound-assisted extraction with a 50% of ethanol with hydrophilic nature. The second one is a CO2 hop extract obtained by supercritical CO2 and with a lipophilic nature. What they have in common is the same variety, origin, and harvest year. So here we have the characterization of the phenolic profile of these two extracts, and as we can see, the hydroethanolic one presented the higher quantity of phenolic compounds if compared with the second one. But if we look at the main and constituent bioactive compounds, the phenols specific for hop, even if the CO2 hop extract presented a relevant quantity, they were lower than the hydroethanolic one. But the interesting thing is that if we look at the bitter acid profile, the opposite trend can be observed. And this is confirmed by the fact that the kind of extraction technology and the type of solvent can select a classes of compounds in respect to another. In this case, we are speaking about apolar substances, and so CO2 was more able to extract this kind of compounds. The chemical composition of these two extracts reflects also the different antioxidant capacity evaluated by two different assays based on of the reducing power for the FRAP assay, where the hydroethanolic hop extract resulted higher, but with a lower radical scavenging activity. So about the technological functionalities of the two extracts, it is supposed to have a complete characterization for both interfacial and surface properties, but at this time I already done only the surface properties determination. And how we can see from this graph, we can assert that this kind of extract in a specific range of concentration between 0 and 0.1 gram liter have this surfactant agent. So we confirm its foaming ability. So about the second activity, the microencapsulation, now we will focus on the hydroethanolic, which I will recall it's hydrophilic. And last year we live with this first trial with the test of freeze-drying microencapsulation technology using maltodextrin, Arabic gum, and their combination as wall material. And the microencapsulation was able to protect the polyphenol compounds, but not alpha and beta acids. So this year, what I think is to use beta-cyclodextrin as an alternative carrier, because thanks to their chemical structure can be able to form inclusion complex and try to entrap this alpha and beta acid with their apolar nature and try to protect them. So what we did is to repeat again the same experimental trial using freeze drying, but this time using beta-cyclodextrin in the formulation of the carrier. And at the same time, I decided also to test another encapsulation technologies, which is spray drying, that even if use a higher temperature compared to freeze drying, it's time and cost consuming. Before spray drying, I did a pre-optimization of the process, evaluating two different inlet temperature, and after the physicochemical characterization of the powders, we decided to select 150 because enabled to produce powders with the lowest water activity, moisture content, with the highest phenolic compounds retention, and also FTIR spectra confirmed a higher interaction among bioactive compounds and the wall material. So these powders were produced and a storage stability test has been performed in order to assess the effect of the microencapsulation. And these powders were chemically characterized at each storage time. Here I will show you some results. For example, here we have the xanthohumol, which is one of the main polyphenol present in hop. And if we compare, based on keeping constant the carrier, the freeze drying, as expected, showed best performances in retain and also during storage for the sample. But if we look at the wall material effect, both in the freeze drying and in the spray drying, effectively beta-cyclodextrin helps to protect these bioactive compounds. The same behavior was for the alpha and beta acid content, so higher freeze drying performance than spray drying. And looking at the effect of beta-cyclodextrin, they help me to protect alpha acid, but unfortunately, again, not beta acids. About the CO2 hop extract, here we have with a lipophilic nature, here we have the explored possible strategies that I performed in teamwork with my colleagues. So the ball milling technologies, which consist in a solid-solid encapsulation, since it's lipophilic, it's difficult to disperse in water solution, or to form emulsion, to form core shell microcapsules by drilling or by spray drying. Those are the future activities The idea is to evaluate, of course, the sensorial properties of this ingredient, because we can recall with this bitter taste could be a problem. And the idea is also to apply the hop-based ingredient in the food system and evaluate the aspect of the in vitro digestion, bioaccessibility and stability of the bioactive compounds at the end of the digestion, and possible cytotoxicity. These are my research outcomes, and thanks for the attention.
Thank you very much. Very interesting. Any question?
Okay, thank you for presentation. Have you a program to investigate some other bioactivities
Right.
besides antioxidants? No? You see a lot of bioactivities, and you intend to focus on some other activities, and in which system? In vitro, ex vivo, animal
So,
models?
because we start from, these are the first studies of the hops microencapsulation. So, in literature, I didn't found any kind of other microencapsulated hop extract. At this step, of course, we will start from the simple things. So, start from the in vitro, and then maybe we wish to arrive to the in vivo system. We are thinking to use this kind of ingredients. The possible application could be different. Even in, let's say, sauce like mayonnaise, or also in bakery products. So the future activities that we wish to perform is to try to apply this kind of ingredient in the food system, and of course analyze all the effects that it have. For example, also it could be interesting also to evaluate the microbiological aspect. So because it is well known that hops act also as antimicrobial agent. So the idea is going on to have a sort of characterization from any kind of view for this ingredient.
Any other? I have one curiosity. Actually, somehow recall what Aldo was saying. Clearly, when you look at the antioxidant, right, the reducing power going up, the other one, the TEAC, that probably is lipophilic. I don't know which one of the two do you use it, probably is that. So clearly, that's an impact of the different aspects, the same, two different phases of the same story. But actually, what you're going to do, it's absolutely interesting and important when you are going to test accessibility, metabolites, and so on. That's why it becomes crucial what Aldo was saying, because you cannot just rely on the fact that you increase somehow the content of polyphenol.
Yes.
You need to, the fact that you pass through the gastrointestinal tract, so you mimic the digestion, that's quite crucial. Nobody do it, and it's really important. So the advice is really, whenever you have the final products of the digestion of all the treatment, also bioaccessibility is not really reliable, but can give you an idea. But together with simple assay, that is not just the antioxidant capacity, but also moving to a little bit more, not complicated, but a complex system like could be cell, also, if you just look to redox status, the chlorofluoreshane, it's easy, no costing assay, anyone. So, I think you should have both data related to what happened after the digestion in terms of metabolites, bioaccessibility, but also how these affect the functionality with simple assay. Okay, any other? Yeah.
Very nice. Okay. I have a loud voice, so I don't know if I can avoid the microphone for all the others. So, just a curiosity that maybe you already explained answering to Professor Corsetti. The curiosity was in which kind of food system you think you can apply this kind of functional ingredients, and you say maybe like mayonnaise, and then you mention bakery products. So my question is, do you think, I know that you have not already done that. But do you think it was maybe more useful to add these functional ingredients in a raw food that do not need to be cooked? Or also there is another problem that comes out, that is the cooking of food. So after cooking, do you think that this functionality, this technological functionality can remain, can be affected, can be erased? So, I mean, just...
Oh, okay, the challenge is this, because microencapsulation in theory can help to also to protect these bioactive compounds, and the right carrier and encapsulation technologies need to be selected also based on this, because selecting the right combination, you can have a final ingredient that maybe can resist also to high temperature. We perform a storage stability test at 50 degrees for this reason, not only to make a sort of accelerated shelf life, but also to test what happens at high temperature. Of course, 50 degrees are a mild temperature if compared to a boiling system. But from literature, it's well known that, for example, hops is used during the boiling part, during the production of beer. These compounds can in some way resist to these high temperatures, but they change their form, they form their isomers, which have at the same time functional properties. So yes, could happen, but it does mean that could be negative.
Thank you very much.
Okay, yeah.
I just have a curiosity because I imagine that you did the supercritical carbon dioxide extraction out from the university.
No,
so
this is a commercially available.
okay, so it's already available. I mean,
yes, we bought this kind of extract. The
okay.
second one we produce by ourselves,
okay,
and comes from a series of optimization performed in our university.
Okay.
The second one is commercially available because it's a sort of way how brewers can use hops instead of using cones. They use directly this kind of extract, but it's very difficult to handle because it's like a resin. So this is why also the encapsulation can help also for this, because handling a powder is different to handle a sort of sticky extract.
Okay. And also, of course, I know that you are making some evaluation of antimicrobial activity, but just I just want to make to highlight, I mean, this kind of activity, because also in the past we work with hope essential oils, but also with extracts with very good
yes, if
results.
I'm not wrong, we test also some encapsulated already. And we saw that they have a very strong activity against the listeria, so it could be interesting.
I just suggest you to not overcome
to
this point, because thinking about potential applications, you can also use it alone or maybe with other ingredients also to exert an antimicrobial activity that could be useful also for the stability.
Yes,
Thank you again.
Thank you very much. Move on. Rossetti, enological versatility of Starmerella bacillaris in wine production.
Hello. Did you see it?
Yes.
Okay. So, today, good morning everyone. Today I want to talk about my research project that titled Enological Versatility of Salmonella bacillaris in Wine Production. For this brief overview, I want to start with the aim of my research project, that is the exfoliation of Salmonella bacillaris in Oms Candida zemplenina in still and wine sparkling fermentation. Moreover, the efforts are focused on further investigation of the effects of cells' lifestyle on wine characteristics. Regarding cells' lifestyle, we can talk about planktonic cells that grow as free in suspension instead of sessile ones that are together enumbered in a self-made matrix, and this state is lead with adhesion properties and biofilm formation. So, contextualizing why I work with Starmarella bacillaris. Starmarella bacillaris is a non-Saccharomyces species that commonly associate to grapevine environment. And non-Saccharomyces species are commonly found on grape berries and show different enzymatic pathways instead of the Saccharomyces cerevisiae one. So, could allow the production on wine with different traits instead of ones obtained with poor culture of Saccharomyces cerevisiae, and could be a good strategy to modulate the wine characteristic in order to achieve wine, novel style of wine, and intercept the consumer acceptance. So, among them, Starmarella bacillaris under the spotlight of research academia could show interesting enological characteristics. For example, when used combined with Saccharomyces cerevisiae, could allow the obtain wines with lower ethanol and higher glycerol content. Regarding the adhesion properties, we starting from the evidence in literature that microorganisms in sessile state shows different metabolism compared to the counterparts in planktonic state. So the question was how this metabolic shift influence the characteristics of wine. In order to better understand if this metabolic shift could be a good strategy in order to modulate the characteristics of wine. Before go ahead, I want to make a preamble on my strain. All strain used during this research project belong to our laboratory. The strain were isolated from organic grapes and mass of different Vitis vinifera cultivars of Abruzzo region in order to valorize the biodiversity of this region, and all strains used were previously characterized for conventional and non-conventional enological characteristics, and the ability to forming biofilm on a biotic surface was evaluated too. For example, here we can see an image obtained through confocal light scanner microscopy in which some Starmarella bacillaris were testing for the ability of forming biofilm on stainless surface. And we can appreciate that the ability to forming biofilm is a strain-related ability. So this fact had to be considered when we selected yeast for wine strain for wine fermentation. So what I'd like to do today with you is try to deline a road of my research timeline, and starting from the beginning, the first activity was focused on the evaluation of the effect of coenoculum of a flocculent Saccharomyces cerevisiae strain and Torulaspora del Burechio Starmarella bacillaris, in order to verify if it could possibly shape the enological and aroma characteristics of sparkling wines obtained with Champenoise method. For doing that, main enological parameters and autolysis and volatile and sensory profile were determined. Regarding the autolysis outcome, the presence of non-Saccharomyces improved that. And for concerning the volatile profile, the quinoculum Saccharomyces cerevisiae and Starmarella bacillaris exerts a stronger effect in the definition of sparkling wine volatilum than Saccharomyces and Torulaspora del Burechio ones. Moreover, regarding the sensory profile, sparkling wines obtained with Starmarella bacchillaris showed the high score for descriptor for spicy, bread, crust, freshness, and floral. So the activity 2 and 3 were aimed to start to evaluated how the cells behavior, the strain behavior, and the adhesion properties could influence the characteristic of wines. So for this activity, we want to evaluate it if the adhesion on oak chips of Starmarella bacchillaris strain could have some effect on volatile profile and main physicochemical characteristics. For doing that, 10 Starmarella bacchillaris strain isolated from Montepulciano d'Abruzzo grapes were used, and the strain that showed the best ability to adhere on oak surface was selected, and where fermentation in lab scale were performed and compared with Starmarella bacchillaris in planktonic state and pure encoinoculum of Saccharomyces or Saccharomyces and Starmarella bacchillaris. So the results highlight that Starmarella bacchillaris adhered to oak chips as release specific compounds such as methyl vanillate, ethyl isobutyrate, and ethyl isovalerate. And regarding the main physicochemical characteristics, the trial F, in which Starmarella bacchillaris was adhered on oak chips, shows the highest glycerol content. Related to this result, in the third activity, we want to investigate the potential of Starmarella bacchillaris adhered on oak chips in order to improve the enological characteristic of Trebbiano d'Abruzzo wine. So starting from a grape generally classified as neutral. So for doing that, pilot scale fermentation were carried out and was evaluated the effect of sequential and coinoculum of Saccharomyces cerevisiae and Starmarella bacchillaris strain, and was evaluated also in trial W6 and trial W5 the effect of Starmarella bacchillaris strain adhered on oak chips. So regarding the volatile odor compounds detected, we found qualitative and quantitative difference, and the principal component analysis differentiate the one obtained in three groups. In the orange one, we find the W5 and W6, so the trial in which Starmarella bacchillaris was adhered on oak chips, and moreover, we evaluated also the colorimetric traits of wine obtained according to CLab colorimetric standards, and the results highlight that in the trials W6 and W5, in which Starmarella bacchillaris was adhered on oak chips, there was an increase of B components of the yellow component. For the last activity, we want to evaluate it starting from the evidence in literature that also strains that were pre-adapted in sessile state. So when the cells were attached on a surface and then detached, could maintain the metabolic shift. So we want to evaluate if using Starmarella bacchillaris strain biofilm detached, we could shape the chromatic characteristic of Montepulciano d'Abruzzo wine. So we carried out lab scale fermentation, and we evaluated the effect of 10 Starmarella bacchillaris strain. The results show an increasing of anthocyanins, polyphenols, and glycerol content, and in order to evaluate if this difference could be perceived also by human eyes, we calculated the color difference in syllab units between the ones derived from inoculation Saccharomyces cerevisiae and Starmarella bacillari, grows as planktonic or biofilm detached cells. 6 trials show value higher than 3. So we can say that this difference could perceive in color trace, could perceive also by human eyes. So talking about and resuming the project outcomes, we can say that for activity 1, the sparkling wines obtained with Saccharomyces cerevisiae and Starmarella bacillari were well differentiated. For activity 2, activity 3, the surface adhered behavior should be considered for the improvement of strategy, for development of organic wines, and eventually obtain of a style of wines. And this is true for Montepulciano and Trebbiano d'Abruzzo wine. And regarding the biofilm detached cells, promising results were obtained, and the wines obtained with biofilm detached cells, Starmarella bacillaris, show an increase in red tint than those obtained with planktonic cells. So I want to conclude talking about where we are going. So basically 2 activity, the phase 1 that is related to the activity 4, and I'd like to call this activity sniffing the difference, the role of Starmarella bacillaris biofilm detached cells. So we want to perform pilot scale vinification and evaluated the effect of cells, biofilm detached cells on Montepulciano d'Abruzzo wine volatilome in order to verify if it's possible to use this biofilm detached cell as started during vinification, and if it's a good proposal for wineries. And the last one, try to figure out and explain why this metabolic shift could happen, and in particular finding the difference in gene expression between planktonic SSL cells through the evaluation of transcriptome performing RNA sequencing analysis. Thank you for the attention.
Thank you very much, Alessio. Any question? Okay, it was clear. I don't have any question. Donato, do you have any question on wine, Trebbiano, Montepulciano? Was clear. Thank you very much.
Thank you.
Let's move to Trasmundi. Study and characterization of plant growth rhizobacteria to improve agricultural productivity and sustainability, a challenge against climate change.
Okay. Good morning to everyone. As the professor said, my activity are based on the study and the characterization of plant growth promoting rhizobacteria to improve agricultural production and sustainability. This is a challenge for climate change. So a brief introduction for about my activity, but it's important to specify the soil. In precision, the biological fertility of soil. The biological fertility of soil can be defined as the quantity of living organisms in the soil and their activity potential. As we can see in this figure, we can divide the soil in 3 respective parts. So the aerial part, this is the aerial part where we have the plant, and the rhizosphere, the endosphere, and the phyllosphere that we can found the microorganism. When we discussing about microorganism, is inevitable, and I think also mandatory to mention the soil, because if we think if we take 200 grams of soil, we can find 0.5 grams of living organism, and this is very important for searching bacteria, fungal, and protozoa. But it is also important because for the study of the microbiome. I don't know why we cannot see this figure, but I can explain briefly. The microbial community have an intelligence that can we interact with the soil. So we can focus our attention and we can consider the microbiome and also the microorganism as a sentinel for stress and also climate change. So, in which mode we can think about the application of our microorganism in agriculture for agronomic application, environmental application, and industrial application. The topic is the same, the agronomic application about the mitigation of biotic and abiotic stress, and at the end, the industrial application about the development of biofertilizer or biostimulants. The aim of my project. I divided the project in two respectively parts. The first part is to isolate and observe the microbial community, but focus on the concentration and the presence of PCPR in two horticulture crops, as courgette and tomato, managed according to no-tillage processing. Identify the species and concentration distribution of possible PCPR by analyzing the difference in their physiological profile, and analyze the different biological mechanisms that join in the soil and under biotic and abiotic stress. The second part is still in progress, but I can say some things about the study of isolate under salinity stress condition in vivo and in vitro during germination and early growth, and identify a consortia of these microorganisms for have a synergic approach and a synergic effect on the plant. Because it's important to underline that this microorganism is difficult that can be affect only in single, but is good for test in consortia. In brief, the material and method are very brief because I isolated this microorganism in Monsampollet, Tronto at Crea in the Marche region. For first, we collected this sample on the summer on 2022, and we divided this sampling in organic tomato and no tillage, and in organic cucurbita no tillage. We did analysis as pH, nitrogen, organic carbon, and sugar. And after that, we identified this PGPR as potential PGPR, because we don't know if are PGPR. So the isolated was evaluated by the producing of indolacetic acid, siderophores production, and phosphate solubilization. And also we did the assessment of the potential PCPR antagonistic activity versus Alternaria alternata and Fusarium oxysporum. A brief result about the chemical analysis of soil. Here I want to concentrate the attention about the concentration of microorganisms in the two theses, organic cuc NET is organic cucurbitaceae, and organic tomato. We can see that the concentration is higher in org cuc and NET. And we have other parameters that can differentiate the microorganism, but we can correlate the concentration of microorganism on every parameters, also the electrolyticity, conducibility, carbon, and nitrogen. These are our isolate, but this is only the first stage, because here we have the concentration of in log, but we start from a total of 147 bacteria, but only 28 reported the characteristic to be considered our bacteria PCPR. This is a table about our identification of 28 PCPR, and here we have a PCR that can show that with this PCR we can understand the distribution of our microorganisms. We can see in orgtom and T we have a major biodiversity of this microorganism. Here we have a radar plot for the evaluation of the potential of plant growth characteristics. So here we can see the siderophorus unit in the upper of this triangle, EAA indolacetic acid concentration, and phosphate solubilization. Okay, here we selected the microorganisms that have a major plant growth promoting potential. So we found 5, 6 microorganisms, but every 28 are interesting. So in the first stage, we take an assessment about the potential of antagonistic activity, and we try in dual culture with Alternaria alternata and Fusarium oxysporum. Here we found a positivity activity of only these 7 bacteria. And here we can see the inhibition mycelia growth about versus Alternaria alternata and Fusarium oxysporum. Here we can see Petri plates with in dual culture, and we can see that this microorganism growth very well versus Alternaria alternata or Fusarium oxysporum, and we have inhibition. But this type of assay requests another in experimental as the production, for example, of antibiotics. But one characteristic that we know is the production of siderophores. The second part. So we conclude with about the characterization, the isolation of our microorganism, and we try to test our microorganism in vitro under salinity stress. As I said, the survey is the climate change. So the salinity stress is most important because can damage our culture and upgrade the salinity stress. So we did some assay about halotolerance about the PCPR halotolerance assay in vitro, and the same as evaluated in vitro with culture while sprouts with sprouts. So the first, we try our bacteria in plate in vitro with different concentration of salinity stress, 100 millimolar, 200 millimolar, and after we try in parallel the control with 100 millimolar of NaCl in vitro with sprouts of our sample and 200 millimolar of NaCl. We can see that the NaCl can damage our sprouts, and the good results are here because we found that this microorganism, Bacillus altitudinis, can affect positively our sprouts. We can see the difference between the previous slide because Bacillus altitudinis in single can higher the biomass of our sprouts. And also with 100 millimolar of NaCl and 200, we have a very, very, very good results. This is only an example about the root length and the shoot length that we can see very good in another slide, but I selected only one, but we have also other bacteria that have a good results. And we can see the results of previously slide here. We can compare the control with Bacillus allotolerans, and we can see is very evident the root length is measured like the control, and also the aerial part or hypocotyle. The activity there are still in progress. We want to do a spectrophotometric the determination of TPC, TFC, chlorophyll, and antioxidant activity of our sprouts. This is important for check if our microorganism can be also a higher concentration of TPC or TFC. This is important to maybe to development of sprouts that can be considered as a healthy food with a good, for example, a good concentration of TPC or TFC, for example. Another thing is to make, as I said before, to develop a consortia and test with species that are hypersensitive with salinity, as lettuce. Further investigation we carry out on the biochemical mechanism to the identified PCPR, and in vivo test we will integrate the salinity with water stress. So what are the conclusions? So we found that the management strategies have a good effect with the microbial community, but specifically about the PCPR composition. So we have we have say that have a direct influence on the composition of soil and plant-associated microbial communities. For second, in our study, we identify 28 potential PCPR with different characteristics that will be tested. Bacillus altitudinis and allotolerans have a very good effect on our sprouts and under versus Fusarium oxysporum and Alternaria alternata. As I say, further investigation will be carried out on the biochemical mechanism. Further in vivo tests integrating with water stress, and what is the perspective of the future. Our goal is to develop a PCPR consortium for a formulation to integrate in agriculture system, for maximize the biomass, the product, but also to survive the climate change and can be more resilient and sustainable agriculture for our culture. This is a brief slide with my publication and participation in conference. And thank you for the attention.
Thank you very much. Any question? Yes.
Ah, sì, no. Yeah. Thank you too.
No, I have just a curiosity. Are you planning also to characterize the enzymatic profile of the
Yeah,
products?
this is an idea.
You know that there are
Yes, this is an idea for the next project, and we consider them. Yes, because it's important to understand also about the sprouts. No, as I say, we have to characterize the polyphenol profile, but also we have to understand the enzymatic activity, because we don't know nothing about this, but also the data that we have in literature. So this is a very good idea. Yeah.
Okay. I have just an advice for you. Did you realize that they were not following you? The audience was not following you.
No?
No, you were looking to the screen.
Yeah.
You don't have the touch, the high touch with them.
But I think that is the answer of the...
Right, no, that's why we do this. But, you know, this is an aspect that you have to consider. Not just you,
okay.
you know, with you was evident.
Maybe I receive my performance to...
Right, no, the point is, when you go on that floor, you have to look at them, catch them, and bring with you until the end of your presentation. because you were absolutely out of the focus. You were not looking them. You were looking the screen.
Yeah.
So after a while, considering also the data are not easy to follow,
Yeah.
they were just doing whatever they want.
Okay.
So that's, you know, it's
I
not
will
just
follow.
matter of presenting the slides, it's also a matter of catching your audience, keeping with you until the end. That's focal for a presentation. Okay, thank you very much. Let's move to the last one, last but not least. Eugelio, development and application of mass spectrometry approaches for food safety and quality.
Good morning, everyone. I will. Okay. Can you see the presentation?
Yes.
Okay. My PhD can you see that?
Yes.
Okay, perfect. My PhD project is about the development and application of mass spectrometry approaches for food safety and quality. The mass spectrometry, first of all, is a powerful analytical technique that can be applied in different fields, such as in food science. And during my PhD studies, I had the opportunity to work with different approaches in both low and high resolution mass spectrometry, especially for the study of bioactive compounds and contaminants in many food matrices, in particular lupins. The first approach I will talk about is the targeted, and here are reported some examples, some research developed in our laboratory. And the targeted involved, first of all, the development of a suitable extraction and cleanup method for the decrease of the matrix effect. It can be performed in multiple reaction monitoring in low resolution mass spectrometry. The advantage of this approach is that it's very useful for the identification and also the quantification of a selected analyte class. And with the use of standard references, of reference standards. So I explain you, I report you as example this work on food safety and regarding the development of a method for the determination of phomopsin A in raw lupin samples. The phomopsin A, as clearly explained my colleague, is a mycotoxin that is produced by the Diaporte toxica, and it can affect especially the lupin plants. It is very dangerous for human and animal health, but nevertheless, it is not regulated yet in Europe. But according to the scientific opinion of EFSA, it is necessary the validation of a procedure for the identification and quantification of mycotoxin. And so we developed this analytical method to satisfy this aim, particularly focusing on these two steps, with the aim to purify the sample from unwanted compounds and have a best recovery of the phomopsin. The first approach was the use of ultrafiltration and centrifugation to separate the macromolecules from the samples and avoid the cartridge saturation of micro-SPE, because we also finally tuned the different steps of the micro-SPE to obtain a suitable cleanup and enrichment step. And at the end, we obtained the decrease of the matrix effect and a good recovery of the phomopsin. So the method was then validated according to international guidelines and applied on artificially infected raw lupin samples supplied by the colleagues of Food Microbiology Lab and published on JCO. The second approach that I will talk about is the semi-untargeted, that is also performed in low resolution mass spectrometry. The advantage of this approach in comparison to the first one is that in this case it is possible to explore the complete profile of a selected analytical class in a sample, putatively identifying the molecules without any use of standard references. And so, in this case, I will report you this work that regarded the complete fingerprinting of alkaloids for lupin samples traceability. The quinolizidine alkaloids, first of all, are cyclic compounds containing nitrogen that are very abundant in lupins, to which they confer the typical bitter taste, and they are so toxic. For this reason, it's necessary that lupin are undergone to appropriate washing and the bittering process to decrease the levels of these alkaloids and to allow their edibility. But important thing is that the level of these alkaloids can be variable according to different factors, and for this reason, they could be. For this reason, in this work, we propose them as biomarkers for lupin traceability. And so this is the method that we applied on we developed and applied on different raw lupin samples from different regions of Italy. And we were able to identify 27 lupin alkaloids and apply 2 approaches of multivariate analysis with the aim to discriminate the samples according to their origin and on the base of their entire alkaloid profile. And these are the alkaloids identified by the analysis. On the left, we can observe the 6 structures identified and confirmed by the use of reference standards as in targeted, while on the right, we found these other alkaloids, and they are reported some of the 21 alkaloids identified with this new method in a semi-untargeted approach. And this is an example. We obtained the chromatographic peak was detected in a MRM survey scan. And then the correspective MS spectrum was generated. And by the study of this spectrum and the deputization of the fragments, we were able to match it also with libraries and literature, and putatively identify the molecules to which it corresponds. In this case, it could be the angeloeloxylupanin, putatively identified without any use of reference standards, only by the study of the fragmentation spectra. And then, after the identification, a clustering approach was applied to have a general overview and confirming a partial discrimination among the samples. After that, a supervised LSDA cross-validation was applied, demonstrating a synergistic contribution of the alkaloids for the geographic classification, and also confirming the possibility to use these alkaloids as biomarkers for the food traceability. And at the end, the last approach that I will describe you is the untargeted, that will be performed in high-resolution mass spectrometry. And it is the most powerful among the other ones because it allows to obtain a complete fingerprinting of a sample and to explore a wide range of it allows to obtain the simultaneous identification of a wide range of analytes belonging to different classes and markers. And so I will talk to you about now this my last work that was performed during my PhD period abroad at University of Almeria, with the aim, in this case, to study the metabolic differences of raw lupin samples according to their ecological origin and also according to farming practices. So the analysis was performed by Orbitrap high-resolution mass spectrometry, coupled with chemometric approaches to obtain at the end a complete fingerprinting and markers identification. So for both the categories, an unsupervised PCA and a supervised PCA and a supervised OPLSDAs were successfully applied, demonstrating a good discrimination of all the samples, thanks also to the identification of specific markers. The markers were identified through the compound discoverer and CFMAD software, according also to their VIP value, p-value, and their fragmentation spectrum. We can observe as For both the categories, the most important markers belong to the class of alkaloids, highlighted in green, and of lipids, highlighted in pink. Some of them belong also to the class of peptides and amino acids. And now it is necessary to deepen this the study of the correlation of the presence of these compounds as markers with the ecological origin and also with the farming process. So, in conclusion, we can affirm that this technique, the LCMSMS technique, is very powerful for investigation in food quality and safety because it allows the quantitative analysis of bioactive compounds and contaminants in a targeted approach. It allows to explore the complete profile of a sample by semi-untargeted or untargeted analysis, both in low resolution and high resolution mass spectrometry, and coupled with chemometric approaches, it allows identification of markers for food authenticity and traceability. So for the future, I hope to deepen these untargeted metabolomics approaches based in particular on high resolution mass spectrometry and chemometrics tool, exploiting in particular open source programming languages. Here I show you the publication during my PhD studies and the conferences on which I attended, and these are the work in progress. Now I want to thank my analytical chemistry group of University of Teramo, the group of Sapienza Università di Roma, e Università di Almeria. And I want to thank you, all of you, for your time.
Thank you very much for your excellent presentation. Any question? Was not easy, yeah? Yes, please.
You can hear me. I have a question about the traceability study. I would like to know if your statistical analysis was based both on now maybe you can hear me better. If your statistical analysis was based on the compounds identified in the target approach,
so this is the second work because I didn't hear very well.
Okay, my question is your statistical analysis of data for the traceability and study was based on both data obtained from the target approach and semi-target approach, because I guess in the semi-target approach you only putatively identified compounds, but you didn't quantify the compounds. So you were able anyway to obtain robust statistical evaluation of data for traceability purpose.
Yes, because in this case we didn't do. I don't know if I understood the question, but I try. In this analysis, we didn't perform the quantification. We just confirmed the presence of these compounds of the 6 target analytes with reference standards because the quantification was already performed with the previous work. So it was already validated and so on. In this case, the objective was to integrate the peak areas, normalize them, and so they were all the same, they were normalized. So according to this factor, then it was applied the statistical, they were applied the statistical models. First of all, the cluster analysis with heatmap, and then the PLSDA. And in this case, the fact that we wanted to demonstrate is that if we apply the PLSDA on only the 6 targeted analytes, as in the previous work in which we work only in targeted, it is not too much possible to classify the geographical origin of the samples, while with the synergistic contribution of these new molecules, so having a complete profile of the lupins from a point of view of alkaloids, it is possible to use them as biomarkers. I don't know if I answered you to the question.
Thank you very much. So we finish this first session. We move to the other one. Professor Del Carlo had a problem. Send me an email tonight. So we will replace with Marina. Ramal Sanchez, you know everyone. She's not of the Collegio of the Food Science, but that's what we found it. No, I'm joking. She's good. Please.
So, good morning, everyone. I will do my best pronouncing the names. So, the first one is Bernardan Monhosu. Okay? Sustainability, Market, and Territorial Enhancement. So, please, the floor is yours.
Okay. Hello, everyone. Okay, my thesis is on sustainability, market, and territorial enhancement. So my name is Bernardet Belosimonsu. I think I'm the only one economist here. Everyone is on biology, microbiology. So I work with Maria Angela Perito and Silvia Codoroni. I work also with the CREA, so my external tutor is Alessandra Pesce. Okay. Okay, what I'm saying is plan on this. I will talk about the structure of my doctoral thesis. I will talk about the work in progress and the published one, and at the end, the future development of my thesis. Okay, my thesis is on sustainability, market, and territorial enhancement. As you know, many aspects of sustainability remain vague and unexplored. The main of the research is to better understand the connection between consumption dynamics and the farm remuneration. So we divide the thesis on two aspects. The first one is consumption reflected by the demand, and the second one is production reflected by the supply. So we'll talk about consumers' behavior, and then the company structure and the market adaptation from consumers. Okay. So the first part of the thesis is the one that most developed. We work on two projects, two papers. The first one is sustainable food behavior in France and Italy, the influence of consumer characteristics. This paper is almost done. We are working on it. And the second one is the European consumer attitude toward ethnic food, the case of dead food. This paper was already published. Okay. You can see that industrialization, urbanization, economic development, and market globalization had led to rapid change in diets and lifestyles. Food preferences in Europe have rapidly changed in terms of taste and lifestyle, and the mindset of how we eat had changed. So we have a difference in a modern diet and traditional diet. So we have the paradox between the globalization of taste, where people want to eat food from all around the world, and food sovereignty, where people want to eat what produced close to home. So on this, we have this paper, the first one, who is sustainable food behavior in France and Italy, the influence of consumer characteristics. To work with this paper, we do a survey. We have a representative sample of population according to socio-demographic conditions in France and Italy on 2,000 individuals. So the variables we use are socio-demographic characteristics, health quality and nutritional aspect, and food attributes. Socio-demographic characteristics are already known, that the gender, generation that age, well, we turn it in generation, her generation Z, millennials, and the baby boomers, the older one. So basically, we do a econometric and statistic analysis. We use a regression model that's called the ordered logit. This model is used for a discrete choice and helps found robust probability for food consumers. So the results reveal that women and more educated individuals give greater importance to sustainable food attributes and care more about the health quality and nutrition aspect of the food they eat. So this is the literature confirms that women care more about what they eat than men. Actually, that's true, I think, even for me. And the second part of this result is that sustainable food attributes are, and the perception of health quality and nutritional aspects differ across social demographics between the two countries that we use. We use France and Italy. France and Italy are countries that close but different from one point of view. That there are two countries that close to the terroir from the food that is produced directly close to them. Okay, we can see that the way France and Italy people see sustainability is different. We can see that women, Italian women, have more interest in the bio-organic than French women. So this is the paper we're working on. It's almost done. And the second paper is the European consumer attitude toward ethnic food, the case of dead fruit. This paper is published on foods already. This paper is different from the other one because it is on ethnic food. So we'll say more that traditional food, food that that is in Europe is imported. So with this paper, we have 1,134 European consumers from Italy and France also. So we compare basically Italy and France. So it is also the variable are socio-demographic characteristic, psychographic characteristic that those characteristics are mainly focused on how people think, how the state of mind of the people, and the food attributes and countries of origin. Food attributes here are mainly, we can say, brands, variety, also bio, fair trade, the geographical indication, and the country of origin, the region of production. Okay. Here also the model we use is the order logit model. That's the best suite of qualitative data that we have. Okay. So here we found out consumers place a high level of importance on organic, GI, geographical indication, and fair trade attributes, and are willing to pay more for ethnic food bearing this label. So basically, if we talk about an ethnic food, like in our case, the date fruit, the consumers, if we have a food that have the label that is bio, that is organic, that is GI and fair trade, they are willing to pay more for this label than the other food that don't have the label. So we'll do also an analysis on the socio-demographic characteristic, and we see also here that women, the more educated and the generation Z, so the younger people, not us, I think we are millennials here, are more willing to pay for ethnic food, bearing organic, fair trade, and GI label, like I said. So we found out here that when people are educated, they're more willing to pay for sustainable products because these organic, fair trade, and GI label are some characteristics of the sustainability. And the literature also shows that the women, the people who are educated, and the younger people, the generations and the young millennials, are the people that are more interested in sustainable issues. So, also, the country country of origin image and the various certification are important in consumers' food choice, even when the product concerned is an ethnic food that is not cultural product of the country of consumption. Like in Italy, we don't produce date fruits, I think. But even for this type of this type of product, the country of image is important. Like I think Italy will produce more like olive oil, and so people around the world know that when we say that olive have come from Italy, that's the mark of quality, even for wine, I think. So the last part is for me is the future development, and this part is basically on the second part of the thesis, is based on the production, the supplies aspect. So here we see that farms have highly heterogeneous economic and environmental performances. So basically, farms in Abruzzo, Roma, or even in Milan are different from many levels, size, farm remuneration, everything. So they are very heterogeneous. So it is important, So it is important to compare farm performances from economic and environmental point of view and try to understand that the impact of the policy by confronting, we will say, homogeneous farm, so that we can analyze. The farms are already heterogeneous, and then we to make the analysis, we have to use homogeneous farm to analyze this. So the methodological approach we will use here will either be the D&D approach, difference in difference, or spatial econometrics. So we have a database, the FDN database that I'm processing right now. This is a database who have more than 86,000 European farms with around 11,000 Italian companies. I found that Italian companies give more variables than anyone else because basically for the country we have around 1,000 variables, but for Italy we have 250 variables for Italian country. So the time series here is over 2008 to 2022. And this survey come each year, so next one we'll have another one. So we use, the variable we use here are like econometric characteristics, like the remuneration of the farm, the fertilization, expenditure, and also social demographic characteristics. This is for the owner of the company, so the gender, the age of the head of the company, and the environmental geographic characteristics that are the longitudinal and latitude of the company, where they are located. So with this information, we could do the spatial econometrics. So with this data, what we are trying to do is to analyze the economic performance of Italian farm by comparing urban and rural inner area farm with the use of the farm level data. So this is what we are trying to do. We are on the processing database. We are trying to use it on startup. And if we can manage that, the second part will be to analyze if the participation to agri-environmental scheme increase farm economic viability. You know that in Europe, we all have the CAP, and all the farms are involved basically in the CAP. So the CAP have very evolved and changed, and now we have agri-environmental issue in the CAP. So we can use this agri-environmental scheme to see the remuneration, the performances of the farms, and so the sustainability in the long term. So I think that was all for me. Thanks for your attention. If you have any questions, we're ready to ask. Thank you.
So thank you very much. It was very interesting. We are running out of time, so if someone has a question, it's now the time. Yes.
Correct me if I am wrong, you collected data using surveys, right?
Yes.
Would you consider collecting data about the propensity of the consumer to contexts, like conversation, public conversation, how many times organic word appears, how many times organic word appears in an advertisement.
Okay.
Because if it works, I don't even know if it is feasible.
Okay.
But if it works, you could predict the results of your service, and you can use those kind of data to correlate with the other class of data that is very similar, like the farm database that you are using.
Okay, but our survey on consumers basically did with an agent survey that guarantee our representativity on social demographical conditions. So what you are asking is if we can use on the internet, the social media, see how many people are talking about, or how often do you come the sustainability part of. I don't know
if it is feasible.
I don't know if I think even we have this kind of data, we cannot use that like that because it will be on the internet, the social media, and we can have the representativity. So we can after throw the information on the whole world. So I don't think that's possible. Okay, thank you. Other question, please? I don't know.
So maybe we could discuss at,
Okay.
at the end. Thank you very much.
Thank you. Ciao.
So next one is Rosaria Aloya, and the title is Evaluation of the Exposure to Microplastics and Nanoplastic Associated with the Consumption of Clan Camellia gallina.
Good morning. Good morning, everyone. And my PhD research project activity is carried out at the Zooprophylactic Institute of Teramo in the Department of Bromatology and Residues. And my project concerned the evaluation of the contamination of microplastics and nanoplastics in Clan Camellia gallina by using different analytical techniques, and the evaluation of the risk of exposure to these emerging contaminants related to the consumption of these mollusks. Microplastics are particles made of polymers with a dimension between 0.1 micron and 5 millimeters. They are found in different shapes and colors. When these fragments break down and reach dimension farther below a micron and up to 1 nanometer, they became nanoplastics. The accumulation of plastics in the environment has contributed to raise the interest of the scientific community on these emerging contaminants. It is known that in some species of fish ingest microparticles dispersed in water that can so biomagnify and enter the food chain. Furthermore, according to some other studies, microplastics act as a vector for the transport of other pollutants. Many methods have been applied for the characterization and quantification of micro and nanoplastics. The most used techniques are divided into types. Spectroscopic techniques on the left, as Raman or infrared spectroscopy. They are non-destructive technique, so they give the chemical characterization not losing the physical information like shape, color, and size. When combined with a microscope, this technique reach 20 micron or up to 1 micron when Raman spectroscopy is involved. Turbogravimetric technique on the right, like thermodesorption or pyrolysis combined with the gas chromatography coupled with mass spectrometry, are destructive technique. The morphological information is lost, but they have a greater sensitivity, so even with a small quantity of sample, the nanoplastics range can be reached. Among the most used complex matrices for the analysis of micro and nanoplastics, there are mollusks, in particular bivalves. Given their nature of filter feeding, bivalves act as are considered usually as concentrators of pollutant because they filter a large quantity of water, and they are considered also for the healthiness of a water body, an indicator of the healthiness of a water body. Clam Camelea gallina is a bivalve mollusk. In literature, there is only one study regarding this species, collected in Black Sea, in which 7 types of polymers were found and characterized with infrared spectroscopy. Given the gaps and considering the need to develop a reliable analytical protocol for the characterization and quantification of nano and microplastics, the aim of my research project are the following. Develop a qualitative protocol for the identification of microplastics using Raman microspectroscopy. Readapt the protocol for the quantification of macro and nanoplastics by using another technique, the pyrolysis combined with gas chromatography coupled with mass spectrometry, to collect contamination data of microplastics and nanoplastics in Clam Camelea gallina, and to, at the end, carry out a risk assessment of the exposure for humans to these emerging contaminants related to the consumption of these mollusks. Here, the activity of my research project using the Gantt chart. The time elapsed to date is shown in gray with the activity number 2, the sampling, that will end in June this year, and the activity 3, so the optimization of the protocol with Raman spectroscopy, already developed, of which today I will show you the accomplished results. Briefly, the sampling of Camelea gallina is carried out by the competent authority in the Adriatic coast, included in Marche, Abruzzo, and Molise region. The sampling was planned as I indicated in the slide. Any choice was made in order to ensure the surveillance of the entire area. A protocol for the extraction of microplastics was developed together with the water biology unit of IZS of Teramo. The procedure involved an alkaline digestion of 10 grams of samples, 10 clams more or less, with potassium hydroxide, and the sample was heated at 60 degrees for 24 hours. After that, 2 gravimetric separation without and with a salt for recovering also the microplastics with a higher density like polyvinyl chloride or polyethylene terephthalate. Finally, the filtration made on a polycarbonate filter for stereomicroscope reading and microparticle counting. At present, it was this last part of the protocol that required the intervention for Raman analysis. In addition, a quality control was made by adding polystyrene particle of different dimension, and an environmental and a reagent blanks are made to take under control the contamination. This is the instrument used in the upper left corner, the instrument used for Raman analysis. From the stereomicroscope reading, the particles are labeled as suspect microplastics that were investigated whether to confirm the polymeric nature or not. And any unknown spectrum was compared with the database available with the instrument, database open source available online, and comparing the spectrum with the ones found in literature. Applied the protocol for Raman spectroscopy means to consider different filters and found the one that can guarantee the least interferences with the Raman signal of polymers. This is the result of this evaluation. A polycarbonate filter is made of a polymeric material, so it has an interfering signal. The use is not recommended, especially for particles of a big size. Glass fiber filter, they don't have a background signal, but the filter that we tried had an irregular surface, and these make them hard to read because particles can go under the mesh, get lost, and so not characterized. They can be used to have additional information, but other type of filters needed to be tested, and in fact it is what we have done. Silicon microperforated membrane, they don't have an interfering signal because the silicon have just one signal that fell at 500 nanometers. The filter has a small size than the other that we tried. The other type of filters that we tried, so the reading is rapid, but they have a higher cost, and they required an additional step, starting from a polycarbonate filter, that involves a loss of particle, especially of the one of smallest dimension. Given the results of the filter test, we choose two routes to analyze the first 5 samples of Camellia gallina. The protocol that involved the silicon membrane and the polycarbonate filter of origin, and the one that involved the new glass fiber filter that we test with a more homogeneous surface. That was actually the problem. I'm sorry? How high? Oh, no, I come back. Here. Okay. Yes. I'm sorry, I was going on and I didn't have to. So, the protocol with the silicon membranes, from a polycarbonate filter, we obtained 4 silicon membranes, and a polycarbonate filter was, in addition, was chosen if it contains particles of a big size, like up to 30 micron, that could be directly characterized on the filter. I want to underline the fact that the recovery rate, as I was saying earlier, the recovery rate of the particles of smallest dimension was near 25%, so not so good. From these 5 samples, we found 1 particles, a green filament of Teflon. And in the slide, I show you also the spectrum in which there are different contributions due to the presence not only of the Teflon, but also of carotenoids, so organic material, and the polycarbonate from the background signal. New glass fiber filters were also tested, and we analyzed the 5 samples also with these filters. This study is undergoing. Here I show you 4 out of 7 particles that we found, whose spectra are under investigation. The recovery rate for the particles of smallest dimension was near, for this first batch, was near 70%. In the immediate future, the activity will, of course, include to carry out more recovery tests for these new fiber filters to confirm the results obtained, to analyze all the samples with the micro-Raman, and to readapt the protocol for the analysis with the other technique involved in this project. Thank you for your attention.
Thank you very much, Rosaria. Any question? Okay, so since we are running late,
thank you.
I would like just to ask you to respect, please, the time. Okay, so because we are running late, and it's important to communicate within the time you have been assigned. So, next one is Walter Polilli, and the title is Water Stress in Horticultural Cropping Systems, Assessment Through Spectral Reflectance. So, please.
Hi, everyone. After all this wonderful presentation, I realized that the one that I am about to do is kind of borderline, but I believe it's in line with the spirit of the Gordon conferences, so let's start and see how it goes. Do we agree? We agree. What do? I work with water stress, specifically on developing a monitoring system on horticultural crops that relies on the spectral reflectance. Now, why do I work with water? Because it is important, it is scarce, it is important, it is scarce, we agree, we can move on. In agriculture, there is a bunch of strategies that can be used to reduce the water wastes and to improve efficiency. I focus on the ones that would benefit the most from a monitoring system, from a sharp and clear knowledge of the water status of the crop. I'm thinking about precision irrigation, I'm thinking about smart breeding, and so on, and so on, and so on. How do we get this kind of knowledge? There is a basket, a plethora of techniques. You can divide them in as you like, but the point is that each and every one of these techniques has a criticality, a flow, if you like. Maybe it is a destructive technique, maybe it requires high-end equipment, maybe the information resolution is too low to serve for the scope. None of them is actually eligible to be the holy grail of the methods that should require to be robust. Of course, you can change the crop, you can change the system, the agronomic management. It needs to be punctual, meaning the information needs to be on high spatial resolution. And it also needs to be quick enough, the response needs to be quick enough to allow you to take action accordingly. But moreover, it needs to be scalable. Why scalability? Scalability is something that obsesses me, as you probably can tell by the question that I made a few moments ago. I believe that this is an underestimated characteristic of technological solutions. But history teaches us that there are many examples where the scalability was the driver into defining if a technology would have been able to outpace the others, to annihilate the competition. On those two examples, two suboptimal solutions like the QWERTY keyboard and the telephone cables were the ones with the highest impact on the world, even if other solutions were much better in many fields, but not in scalability. So a competitive advantage in scalability was the driver here. And in many cases, most cases, the secret sauce is the opportunity to exploit an existing infrastructure. Back on track, I promise. That's right. Back on track, spectral reflectance. Can it be the holy grail? Let's dive on it, okay? We have a main hypothesis, which is pretty basic. It assumes that a light canopy interaction event produce an information that is stored into the spectral signature that can be detected and analyzed with powerful machine learning techniques that can decode the information and provide us the useful one, okay? That is the easy part. The consensus is that it's quite feasible. But we want to go on, we want to perform a feature analysis, hoping this time that the much bigger if down there will turn in our favor. Meaning that a smaller part of the spectrum, specifically the visible and the very near infrared part of it, still carries enough information to allow us to simplify the detector and get our secret sauce by exploiting the existing infrastructure of mobile phones. So what is the plan? It's quite simple. We perform multiple experiments on both a controlled environment and an open field. We use one species at the time, it's Cucurbita pepo, while we monitor a bunch of features that are somehow correlated with the water status of the crop. Okay? At the same time, we take the spectral reflectance as well. All of it in a feedback loop where the controlled environment experiment should serve for developing models and test protocols that are then transferred to the open field condition and validated eventually. And all the study is strictly linked to the capabilities of the equipment that in our case is a portable high-resolution spectroradiometer capable of capturing the visible, the near, and even the shortwave infrared at a very high resolution. And moreover, it gives us a bunch of different data acquisition modalities, each with its pros and cons, but more importantly, each allowing us to test different protocols. And more on that topic, we designed our controlled environment experiment to assess how the measurement usefulness is affected by the when, the where, and the how you take the measure itself. So protocols again. We tested the leaf age, we tested the pre and post irrigation recordings, we tested also the temporal distance from the stress induction. And from that, we get strong indication that some protocols are much better indeed. As an example, the young leaf carries the most valuable information, and our ability to predict the water status decay in time along with the stress reaction that the plants deploy, stress reaction strategies that the plants deploys. And we also established that acute, short-term water deprivation is not relevant at all. So our information goes like that, okay? It rises, then slowly decrease. We can move with the open field experiment where we built up 4 treatments by crossing a water input differential through irrigation and a water loss differential through different soil management systems. Unfortunately, it's an open field. The field commands, the field rules. And it was a very rainy summer last summer, specifically very rainy June and May, which coincide precisely with the early phenological stages of the crops, which are quite critical for water stress. But much worse than that, there is a thing that we need to address, and it's fatal flow. Okay, maybe it's not fatal, but I'm over-dramatizing here. A broken link on the loop that I described before. Machine learning requires data that describe well the phenomenon, okay? But not few of them, a lot of them. Not a bunch, not a good amount, a lot. Otherwise it won't work. And I am not talking about spectral signatures. We are plenty of it. I am talking about water status data that are gathered with the conventional techniques that I mentioned before, are destructive, are time-consuming, are equipment-intensive. So we have a problem here, which, again, is a scalability problem. We also have a solution, a potential one at least, which is based on the assumption, on the basic knowledge that plants move. They do move in many ways for many reasons, and in each movement, water is involved because it acts as a media for the transmission of forces between the plant tissues, okay? More than that, the water stress responses act on the osmotic pressure of the cell fluids. That means that by looking at the movement, we could have an idea of what is the water status of the plant. How do we do that? We can install webcams on the top of the crop system and on the side, 3 on the top, 1 on the side, and take pictures, record pictures every 20 minutes. Now, this experiment, every 20 minutes for the whole duration of the experiment, which ended like 10 days ago, so I do not have too much to show, but there is a thing that I want to show you, or at least I want you to see, and this is a preview of the image analysis, one of the approaches that kind of work. I really like it, because, okay, it looks at the younger leaves, search for it, takes it, and take a measurement of the movement of the younger leaves. And kind of work. But the nice thing is that it's fully heuristic, meaning that there is no machine learning involved in this stage. And sometimes it fails, as you can see, and one fail will start a cascade of more failures down there. More work is needed, of course. But it seems promising to me. What do you think? Anyhow, with this, my presentation ends. So thanks for your attention and, of course, for all the fish.
Thank you, Walter. I appreciated the passion you put on your presentation. So any question? No? Okay, so thank you. And okay. Sorry.
The scalability of this system, because for sure you are working with standard conditions.
Yes. It means it's a scalable system that is not transferable, which means it works in controlled environment, but the transferable part is carried on by the spectroradiometer. So our problem is to correlate the spectroradiometer data to the relative water content, surface leaf area, specific leaf area, to proline content. We have too few data of this kind to define the water status of the crop. So we need an intermediate, a media that take a continuous measurement of the water status that we can use to link with the spectroradiometer. Then we can bring the spectroradiometer on the open field, and then we hope that the spectral informative part is in the visible part of the spectrum. It's a moonshot, probably won't work, but it's nice.
Thank you. So next one is Reindorf Boateng. Okay. Innovative strategies to modulate the delivery and food structuring abilities of starch-based ingredients.
All right. Good morning to you all. My name is Reindorf Boateng, and today I'm presenting on the topic Innovative Strategies to Modulate the Delivery and Food Structuring Abilities of Starch-Based Ingredients. And these are my wonderful supervisors, Professor Paula Pitia and Dr. Marco Feita. Presentation outline, background study. We all here, we are consumers, and we are increasingly interested in clean label food products. That's food product that supports sustainable and healthy lifestyle, and also reduce the risk of chronic disease. Starch, as a cheap and abundant polysaccharide, is used in many food applications as functional ingredient. However, in its native state, it lacks the commercial application. And in order to reverse this challenge, it is important to find innovative strategies to improve its structural properties and other technological functionalities. So we have a lot of modification methods. We have the chemical, the enzymatic, genetic, and physical. But for the purpose of this study, we are focused on the physical innovative technologies like the ball milling to be considered to produce the clean label starch ingredient, because it prioritizes natural production of clean labels and also support sustainability and whilst enhancing the technological functionality of the polymer. Ball milling, basically, it uses mechanical forces such as friction, collision, impingement, shear, impact to modify the processing material, consequently increasing the solubility and increasing the stability of labile compounds. But in literature, there's this gap, that is the use of ball milling modified starches with enhanced functionalities for the food production or food application system. So this work tries to address this challenge. So the aim of the project is to evaluate the potential of the ball milling on starches from different botanical origins, their physical modification affecting its technological functionalities for food structuring and development of innovative structures and delivery system. The project is divided into 3 phases. The first phase is the modification of the starches from different botanical origins. The starches that we considered are corn, tapioca, wheat, and potato. And under the ball milling processing conditions, such as the time, we considered 5, 15, and 30, and at a constant rotation speed of 350 RPM. So this, after modifying these starches, we went ahead to characterize their technological functionality, such as the molecular, structural, and physical properties. The phase 2 is to design and develop the ball mill structures. So basically, the ball milling modified starches, we will go ahead to design and develop their structures. The structures such as hydrogels, cryogels, and other colloidal systems. And then we will evaluate their technological performances. The third phase is to use this modified ball mill modified starches to design and develop microencapsulates and structures as delivery systems, and as well as encapsulate core components such as plant extract, individual or mixed bioactive and essential oil. But this one we are yet to define. And afterwards, we determine its technological functionality. So the first one is done. The last 2 phases are ongoing. As already said, the native starches that were considered were corn, potato, tapioca, and wheat. And under this ball milling condition, rotation speed 350 RPM, the time 5, 15, and 30, a balls to starch mass ratio is 23:1. And after obtaining the modified starch, we went ahead to characterize their technological functionality. So here, depict the results. And it is clearly seen that we can see an increasing trend in functional properties, an increasing trend in functional properties with regards to increasing milling time. And how did this occur? Ball milling undergoes a process called hydrolysis, and this hydrolysis under this process called mechanolysis. So this mechanolysis, what happens is that the ball milling impart mechanical shocks to the starch granules, and consequently, it induces this the starch depolymerization and releasing hydroxyl radicals and free amylose, which is available to interact with their environment, that is water. So increasing the milling time increase these properties. And it's clearly seen that we have the tapioca starches, tapioca starch having high swelling power and water holding capacity. And it's very useful for the food processor because we can use this to enhance the textural properties of food products such as puddings, sauce, dressing of salad. And also the food processor can use in the bakery industry to, because of its high water retentivity, you can retain a lot of water, promoting the freshness of the product, and then delaying process called staling of the bakery product. And for the potato starch as well, it's having higher cold water solubility. And it's very useful as well. We can use this potato starch in enhancing emulsification, and as well as using it in frozen products as well. So the oil holding capacity, you could see an increasing trend of oil holding capacity with regards to increasing mailing time. And it's true because naturally starches, they are hydrophilic. So when you modify the starch, you expose its hydrophobicity. So this hydrophobicity interacts with the oil and increasing the mailing time have increase in oil holding capacity. Here we talk about the viscoelastic properties of the starch. And it's clearly seen that the native starches, they have high storage modulus. That is, it measures the elasticity of the starch as function of the temperature. So when we increase the mailing time, we decrease the elastic properties of the starches. And this is because we degrade the starch granules. So, the ball milling induces the degradation of the starch granules. And when such happens, it promotes the diffusion of water into the starch granules and decrease the resistance to shear forces. And this promotes weaker gel formation. And for the gelatinization temperature, native starches have high gelatinization temperature, whilst the malt starch has low gelatinization temperature. And it's very useful for the food processor because if the food processor wants to produce products incorporating core component that is heat sensitive, you need ball mill starches with low heat gelatinization so that you will not degrade your component. The damaged starch, we could see an increasing trend. When the ball milling time increases, we degrade a lot of granular component, yielding more gelling fragment materials. For the thermal analysis, could see that the gelatinization temperature and the enthalpy of gelatinization for the native starches is very high. So when we increase the milling time, you decrease the gelatinization temperature and enthalpy of gelatinization. And this is because of the damaged crystalline structure, which consequently the ball milling promotes the hydration of the granules in the amorphous region. And looking at the last graph, I calculated the intact crystallinity, the amount of crystallinity left in the starch granules, and how is all came about the native starches, the ratio between the native starches and their corresponding milled starch. And you could see that the potato starch, after 5 minutes of ball mill treatment, it has few or less crystalline structure within the granules, which shows that it shows high sensitivity towards the treatment. For the particle size, the corn and potato, they have small particle size, which means that they have high surface area. So increasing the milling time reduces or increases the particle size growth due to agglomeration. And how the agglomeration forms is when the starch or the ball milling induces the breakdown of the starch granules, it releases small size fragments. And these small size fragments, they have high surface area to a mass ratio. And they have high surface free energy that is able to clamp together or adhere to the surface of the large granules as in the process of, due to the interaction of the electrostatic and van der Waals forces, which consequently increases the particle growth. So these images depict the native starches. We have smooth and they have pronounced shape. Increasing the milling time leads to damage on the surface of the starch and irregular shape and particle agglomeration. So, in conclusion, so far, the ball milling treatment improved overall technological functionalities, and the responsiveness of the starches to the ball milling time varied depending on the botanical origin and as well as the processing conditions. So, here, you have short ball milling time, which yielded significant improvement. We don't need high amount of time, so short processing time can improve the starch functionalities. So these results indicate that the ball milling has a potential for modifying starch, resulting in the development of innovative food ingredients with enhanced functionalities. Ongoing studies, amylose and amylopectin content, we are yet to determine, retrogradation of the starches, in vitro digestibility, resistant starch determination, and also design and development of food delivery systems and structures, the hydrogels and cryogels, and we encapsulate the core bioactive component. These are my references. Thank you.
Thank you very much. Any question from the audience? No one. They're really shy. So thank you. Thank you very much. And just to be sure, Alessandra Cornacchia is not here, right? Okay. So, last one, not least. Ece Ozel, innovative technologies and processes for sustainable frozen foods.
Hello, everyone. My PhD project is innovative technologies and processes for sustainable frozen foods. My PhD thesis it's industrial one, and we collaborate with Solis Greenlock Logistic. So why we do freezing? Freezing and frozen storage systems are important and one of the solution for the food waste, because 12% of the global food waste due to not proper temperature management. With freezing and frozen storage, we can extend the shelf life. It's easy to reach and increase the accessibility of seasonal products and due to our changing lifestyles. In this graph, we can see the increased trend of global food, frozen food markets. And one essay held in America showed that sales in frozen products increase compared to fresh products. However, frozen foods come out with a problem, a significant energy and greenhouse gas emission problem. And cooling technologies currently account more than 7% of all greenhouse gas emission. According to International Institute of Refrigeration, did one analysis, and the specific energy consumption is highest in the Italy year 2015. And total current energy consumption in the cold chain estimated approximately 484 televolt hour per year. And what is the solution? Solution is the innovative and sustainable technologies. Some of them renewable energy sources, solar power and wind, electrostatic fields, pressure freezing, ultrasound freezing, magnetic field freezing, electric field freezing, microwave and radio frequency freezing, and isochoric freezing. Isochoric freezing show that it's a significant degrees of electricity consumption and carbon dioxide emission. Today I will talk about benefits of solar power freezers and storage systems. They are environmental friendly because they are using solar energy. Energy independence, especially where electricity is access is limited or unreliable. Their cost saving, first of all, they seem to be expensive for the first investment, but for the long term, they provide long-term savings. And how do they work? Solar-powered freezers work using solar panels, convert the sunlight to the electricity by using photovoltaic technology. However, photovoltaic generators has a problem. They produce energy not constantly, and the main problem is temperature fluctuation here. And it's needed real-time monitoring and control systems to control photovoltaic energy availability. These are the limitations. And this is the real graph obtained from the company. This green line, what we see produced by the photovoltaic energy, and the red one is consumption in a day. When the red red graph is passed through the green one is need energy withdrawal, electricity withdrawal. This is the limitation. And respect all of these explanation, aim is this project to investigate effect of innovative and sustainable technologies, eco-friendly processes and frozen storage system on the quality of foods. Unfortunately, freezing also have an adverse effect on product quality, such as large ice crystals, moisture migration, recrystallization, and due to long-term frozen storage, some chemical damage. We have enzymatic degradation, protein denaturation, lipid oxidation, and loss of vitamin groups. Respect all of them. Activity is carried out at extensive bibliographic research, evolution and optimization freezing process and frozen storage system by monitoring temperature fluctuation in solar-powered freezing, and selected products quality evaluation during solar power freezing and frozen storage. We have two case studies. One is carrots. Carrot is selected due to large usage of in freezing sector. Another one, freezing and frozen storage of fresh hop cones. I will start with freezing and frozen storage of carrot slices. Carrots are chosen Italian local market. They sliced in the specific diameters, and we have two batch. One is a pre-treatment, and second, no pre-treatment. We use blanching as a pre-treatment. Then subjected to freezing and frozen storage. Here we have comparative study in conventional freezing, held in University of Teramo, and solar power freezing held in collaborated company with three different temperatures. How we choose these temperatures? I don't know why we cannot see. We did the differential scanning calorimetry analysis, and we calculate the T prime G to obtain the points of the storage temperature, and it's I can show you here. Sorry. Here, and it's find out -43, and that's why we decided to choose this temperature according to facility of here and the company. Then analysis have been carried out. Fresh and just blanched without any storage. Carrot slices used as a control. Just to see the effect of blanching, pectin metylesterase and peroxidase enzyme are inactivated. As is industry also, this percentage leaving is good because otherwise it will be cooked. This is the time temperature observation. Here we can see exactly the temperature fluctuation due to nature of the solar power freezings, and this is the freezing curve of the carrot slice held in company. This we just see it. And now I just obtained the first data. It's where we just started this analysis. Here I would like to show moisture content and drip loss. In the moisture content, we can see clearly a difference between blanched samples and fresh samples. When we see the stability of the moisture content, we can see in the -45 blanched sample and solar-powered freezing keep the moisture content stable. Drip loss is important parameter for the industry and for also the economical point of view. And we observe that conventional freezers and not blanched samples have highest drip loss drip loss, and this is but something positive that solar power freezing keep the products with less loss of water. Then we have analysis in color, and we can observe here also difference between blanching and the fresh samples, and colors have a different trend compared to temperature and freezing and frozen storage technique. And the mechanical properties. Samples were frozen from the first graph we can observe at solar power freezers at -18 compared to -20, and conventional freezers show higher recovery work, which is related to the elasticity. And blanching show positive effect on the elastic properties of the carrot slices. And second graph, we can observe the positive effect on the recovery work of the blanching. And we did also total phenolic content assay, and time zero blend samples exhibit lower antioxidant activity than compared to fresh ones. However, after 7 days, frozen storage increased the antioxidant activity was observed in blend samples in both freezing techniques. Then 7 days of frozen storage samples blanch and kept in the conventional freezer show highest antioxidant activity across all the temperature compared to solar powered freezers. And this is my second case study, freezing and frozen storage of fresh hop cone. Hop in general use essiccated in the industry. We have experimental idea that how we can keep in the freezing, frozen and frozen stored fresh hop cones. That's why we are just improving these methods. We start to do in University of Teramo, again with 3 different temperature and extended storage, frozen storage times. We have antioxidant activity with 3 different assays, and all the assays has observed similar trends, and still we need to understand why there is this movement in the time set 7 in first week. And we analyze some phenolic compounds in HPLC, and we can clearly observe we have a production of naringenina, and we have a decrease in isoxanthomol, which are the main components in the hop cones. And alpha-beta acids have the similar trend with antioxidant assays. Future studies, obviously, statistical analysis currently going on to understand better the data. Extended frozen storage of carrots of hop currently ongoing. And we have some idea that why they can act in this way. We would like to evaluate enzymatic activity of freshly frozen hop cones and evaluating effect of freezing without any frozen storage of the hop cones. Then we will have another comparative study with solar power freezing and conventional freezing is animal origin product. And these are my side activities, which have been carried out, and upcoming activities. Thank you for your attention.
Thank you very much. Any question for Reche?
Just one observation, because if you go back to the data, it's quite clear that the third point is always that one going down.
Always that one.
Right, so, sometimes we try to find out Nobel Prize idea for explaining, or maybe it's something technical, because it's the only one that for any kind of a marker you are going to look at go down. So probably there was something related to, not even assay, because are different assay, right? Also the
Yes.
capacity total polyphenol. So something happened at that sample.
We-
Because otherwise, how can you explain that any kind of, not just general phenolics, total FRAP, that are general, not identified with molecules, but also every single molecules, you have the drop and the rise.
Yes, exactly.
So, I mean, it's really hard to believe that an ingredient, any kind, disappear, and after a while, appear again. So probably, you have to consider that instead of trying to come up with any ideas, maybe, making a gain or just take out the data, because, it's something that escape your control.
I did many trials. It's always the same. Also here the same. We were thinking there is type of polymerization, isomerization. We need to still discover what's going on there.
How you recreate the molecule? That's the point.
That's the point.
Went down and stay down. Okay.
Yeah, so maybe the problem is not the 7 day, is the 14 day to drop, because maybe all is growing in a time, but then the problem is the drop is not that you have the 7 day point.
Yeah.
So maybe something happens.
Something happens.
Yeah.
It's my future project to find out. Yes.
Thank you very much. So second session comes to the end. It was a pleasure to introduce you all and to listen to your presentations that I found really different, the one from the other. And even if they are from the same field, like small pieces of a big cake. So I hope you will agree with me the fact that this is a really events like today are really interesting, not only for sharing your ideas or your results, but also for learning something different, but that is related to your field. So congratulations and good luck.
So there we go. We just started the last session, and I call here on the floor Federica Montagano. She will have a presentation about the selection of the food-associated lactic acid bacteria and the evaluation of their differential impact on the functionality of innovative, healthy fermented foods. The tutor is Prof. Aldo Corsetti. All right, go on.
Hello everyone, it's a pleasure for me to be here today. And today I'll give you an overview of my PhD project based on the selection of food-associated lactic acid bacteria and evaluation of their differential impact on the functionality of innovative healthy fermented foods. Actually, fermented foods are defined as foods made through desired microbial growth and enzymatic conversion of food components. And in the last few years, for the increase of new dietary trends, as well as for the increase in allergies and intolerances, has led to the reassessment of plant-based matrices, especially cereals and legumes, for their high nutritional impact. They can also act as delivery vehicles for probiotics and other biotics molecules, such as prebiotics, symbiotic, postbiotics, or psychobiotics. Indeed, there are a lot of different functional impact derived from microorganisms involved in the fermentation processes, so they can act on the modulation of gut microbiota, especially through the modulation of several molecules such as short-chain fatty acids, cytokines, or neurotransmitters with beneficial effects like antimicrobial, antioxidant, anti-inflammatory, and immunomodulatory activities. Our preliminary results were focused on the selection of Lactobacillus plantarum strains isolated from different fermented foods for the GABA production in a legume-based matrix. By an enzymatic assay, we have selected 4 strains among 20 for the GABA production in presence of the GABA precursor MSG. Then we verified the GABA production of the 2 selected strain mixture in a synthetic medium, and then we verified the production of the neurotransmitters during the fermentation of a legume-based beverage. All the results shows the different capacity in produce GABA of the selected strains, also when they've been used in combination. So it confirms that there is a diverse functional impact that depends on the characteristic of the selected strains. So based on these results, the idea of my PhD project came from 2 main questions. The one is, what are the effects of the different inoculum strategies? And the other is, what is their impact on the functionality of fermented products? So the aims of the project will be, first of all, the selection of lactic acid bacteria starter cultures, then the selection and characterization of different fermented foods with the optimization of fermentation processes, and the impact of starter cultures on the fermented foods functionality, with a focus on the anti-inflammatory activity by using an in vitro intestinal cell lines, and the evaluation of the effect on the composition of the gut microbiota by using an ex vivo model. Thank you.
Thank you. Any question from the floor? Just a very small one. Is it a complicated analysis to get the GABA from the HPLC analysis? I mean, you have just a protocol, or you set up it ad hoc?
There is a protocol, but we use the different approaches. At the beginning, we use a colorimetric assay and then an enzymatic assay, and with the help with chemistry group, the spectrometry analysis.
Always chemists. Thank you so much. Okay, so we can go on, and the second candidate is you, Antonella Damiano. Okay. So she will present a talk about the toxicological profile and therapeutic potential of oleocanthal and oleacein in zebrafish models with pathology. The tutor is Professor Michela Moreno. Please.
Good morning to everyone. My research project is a toxicological profile and therapeutic potential of oleocanthal oleacein in zebrafish, the neurario models with pathology. The extra virgin olive oil is well known for its nutritional qualities and health advantages. Its beneficial properties are attributed to its unique composition. For 99%, it consists of monounsaturated, saturated, and polyunsaturated fatty acids, and for 1–2% of bioactive compounds. These letters are represented by 220 different substances like hydrocarbons, tocopherols, polyphenols, alcohols, sterols, and pigments. Among other chemicals, the saccharidoids oleocanthal and oleacein are being studied for their potential therapeutic effects against cancer, neurodegenerative, inflammatory, and cardiovascular disease. but their full ability to promote health remain uncertain. This because most of the research present in the scientific literature on the polyphenols of EVA oil has focused on the hydroxytyrosol, tylosol, oleuropein, and less on oleocanthal and olesin. Moreover, only in studies on their protective effects have been conducted. So the research project aim are evaluation of toxicological effects of bioactive compounds of extra virgin olive oil on zebrafish models, and investigation of beneficial effects of the polyphenols in hexam on chemical models of zebrafish. In synthesis, in the first phase of the research, we will characterize the toxicological profile of an extract of extra virgin olive oil through fish embryo acute toxicity test to evaluate lethal and sublethal toxicological endpoints. Additionally, pharmacological, toxicological, and genetic models of pathology we will created and validated to test in the third phase the therapeutic and modulatory effect of the compounds in different systems through evaluation of biochemical, molecular, and behavioral endpoints. Until now, it has been investigated the acute toxicity of green extra virgin olive oil based formulation in natural deep authentic solvents on zebrafish models to identify a sublethal co-concentration to be evaluated for anti-inflammatory properties. The analyzed matrix was obtained with a sustainable method employing natural deep eutectic solvents proposed by Franchosi and other researchers for giving elevated yields of oleocanthal and olesin from extra virgin olive oil. This because these two molecules cannot be easily obtained via chemical synthesis, and their isolation and purification from air are expensive with low yield. So, to test this extra virgin olive oil, we have exposed embryos to 7 concentration of this formulation in Aedes, and toxicological endpoints were calculated. The lethal concentration 50 calculated at 96 hours post-fertilization of embryos was 3.7 milligram per liters. And the lethal endpoints considered were coagulation, lack of tail bite formation, lack of wort, lack of somite formation. In conclusion, the presented data shed light on the toxicological profile of this evergreen extract, indicating possible the potential pharmacological use, and furthermore, preliminary studies on a zebrafish model of inflammation suggest a protective anti-inflammatory effect of the extract and represent an important starting point for further bioactive studies. Thank you for attention.
Any question? Please.
Are you planning to test other models besides the zebrafish one?
Yes, we will test inflammatory models with the use of LPS and copper sulfate, but regarding all zebrafish models and pharmacological models of zebrafish or toxicological model of zebrafish with other molecules.
That's it. Thank you. La Penna. So we have Francesco Reggi. Francesco will have a talk from the title Development and Technological Functionality of Micro and Nano-Sized Extracellular Vesicle from Plant Matrix Intended for Food Application with tutor Professor Paola Pittia. Please.
Good morning to everyone. This project is about the development and technological functionality of this extracellular vesicle from plant matrices. My PhD project is in collaboration with Exolab, is an innovative startup located in L'Aquila that born 5 years ago and is expert to the production and commercialization of product, in particular of cosmetic and nutraceutical product based on extracellular vesicle. So the principal aim of the project is the exploitation of this extracellular vesicle in food product to enhance the quality, safety, and stability. But what are the plant-derived nanovesicles? As we can see to the image to the center of the slide, plant-derived nanovesicles are small lipid vesicles released naturally by the plant cell, and they consist of a double layer of phospholipid, similar to the cell membrane, and contain a various biological molecules, such antioxidant, such microRNA. So the step of research project are 3. In the first, we select and characterize the extracellular vesicle through the physical property, through the nanosite tracking analysis, Z-sizer, that allow us to count and characterize physically this vesicle. So we also do antioxidant property and antioxidant pattern of this nanovesicle. In the second step, we have the encapsulation of this vesicle to stabilize and to apply in the third step in real system, in food matrices. To date, we perform several activities, so extraction in particular and screening of nanovesicle from bergamot and grapefruit. And we try to extract also the vesicle from hop. So I optimize the extraction protocol of nanovesicle, characterize physically through various methods. The future perspective of this vesicle and of this project is the selection of the better source of this extracellular vesicle, and so we continue to microencapsulation experiment. Thank you for attention.
Any question? Okay, that's it. Thank you.
Thank you.
Next candidate is Chiara Bravo Trippetta. She will present a project about the role of selected foods with different nutritional composition in modulating the anti-pro-inflammatory response in vitro and in vivo, and I am the tutor.
Thank you, Professor, for the presentation. Good morning, everyone. I will share with you my research project. Inflammation is a physiological response of the immune system against different factors. These factors can produce an acute inflammation or also a chronic inflammation, becoming crucial in many diseases. Diet plays an important role in many diseases and also in the inflammation. At the moment, there is not any scientific approach that determine whether a specific food is anti-inflammatory or pro-inflammatory. For this reason, this project seeks to understand the link between food and inflammation. The aim of this project is to test food and recipes for potential anti- and pro-inflammatory effects with different approaches, in vitro and in vivo. We are going to start to identify food and recipes with potential anti- and pro-inflammatory effects. We are going to select different food from all the food categories. Then we are going to develop and execute an in vitro protocol to food digestion. We are going to use the InfoGest protocol that simulates the 3 phases of human digestion: oral, gastric, and the intestinal phases, obtaining a digesta. We are going to test this digesta with the intestinal model using 3 kinds of cells. The intestinal cells CACO2 and HT29MTX-E12, and the monocytes THP1 cells line. And we are going to measure the cytokines released from these monocytes. These cytokines are related with food and the nutrition. Then with these results, we are going to create a scoring system to classify food depending on their inflammatory effects. And we are going to use an algorithm and also a big data, obtaining the NII index. Moreover, we are going to perform an observational study to assess the anti-inflammatory effects of different dietary patterns, and from the database, from different studies already done. And we are going to score each diet. Finally, this project also includes an interventional study to assess the anti-inflammatory effects of lupin seeds. Participants will incorporate lupin seeds into their diet to assess the inflammatory marker. And we can see if the inclusion of this food can modulate inflammation compared to the control group. Finally, this study is expected to provide a comprehensive assessment of the postprandial anti-inflammatory effect of a specific food. And also, these results obtained will lead to identification of food with potential anti-inflammatory properties to be included in a personalized diet, and also identify food that should be limited in diets. Thank you.
Any question? Okay, that's fine. Thank you.
Thank you.
Francesco Della Valle? C'era una domanda, non l'ho vista. Yeah, go on. Sì, statico. Francesco will present a computational mass spectrometry approach for the traceability and sustainability in agri-food chain, and the tutor is Prof. Michele Del Carlo. Please.
Thank you. Good morning to everyone. I'm Francesco Della Valle, and I'm a PhD student in food science in the group of analytical chemistry. And the project is about the computational mass spectrometry approach for the traceability and sustainability in agri-food chain. The project is a part of PNRR Mission 2 Component 1 about the development of a suitable agri-food chain, improving environmental performance and farm competitiveness. And the aim of the project is to develop an informatic tool for the traceability of primary production in order to allow companies and third-party organizations to trace the made in Italy products. For this purpose, it will be necessary to evaluate different biological activity and metabolic profile of different raw products. In particular, it will be necessary to develop an analytical approach for the determination of non-conventional biomarkers, for example, oxidative stress markers in plants, for the process monitoring and farm variabilities. So, what is traceability? Traceability refers to the ability to track and trace the movement of the products and their attributes through the supply chain. It involves capturing and recording information such as origin, production, processing, and distribution of food products. How is it possible? Is it possible using different analytical approaches, in particular targeted and non-targeted approaches? For example, for the low-resolution mass spectrometry, we can use multiple reaction monitoring as a targeted approach, but also an information-dependent acquisition coupled with an enhanced product ion for a semi-targeted approach to identify different classes of compounds. But I think the most important is the untargeted approach for the biomarker discovery, so to discover other classes of compounds. We have different bioactive compounds. We can classify them in two groups: conventional biomarkers, for example, polyphenols, alkaloids, phytosterols, and so on, and non-conventional biomarkers, for example, phytoprosteins and phytofurans that are oxidative stress plants markers. After that we collect all this information, we can proceed with the data processing. The data processing should be carried out in 3 steps, and it is very important to build a model of classification and pattern recognition. So we have the data pre-processing, that is the procedure for making raw data into suitable form, so it's ready for the machine learning. After that, we can perform univariate and multivariate analysis for the model building, so applying supervised and non-supervised approaches, and then the data assessment of the model using RStudio or Python, that are open source program language, and it is an open source informatic tool for the traceability of the primary production. There are many future perspectives, in particular to identify the correlation of molecules to several metabolic conditions in plants with geographic origin and environmental characteristics. The effect of raw materials will be evaluated in form of the antioxidant activity and resolving inflammation response that will be correlated with the environmental and geographic origin. And then this analytical tool will be eventually based on the obtained data with the aim to trace the geographic and environmental origin of food commodities, and the whole will be used to develop a script based on original open source programming languages that can be used and shared in the food industries. Thanks for the attention.
Any question?
Just a curiosity, the resolving inflammation response, because you target, target, so maybe compounds and so on. So I assume that you will assess the geographic correctly and looking at the graphical origin, cultural, blah, blah, blah. But for the inflammatory response, what are you looking? Speculation or a test?
No, it's a test on in vitro and in vivo models. For the in vitro models, we think about that to use, for example, Kakotsu cells, or in vivo models like zebrafish model.
Okay, thank you. The last candidate, Shazia Akram. She will have a talk about the cheese mold spoilage, strategies and new trends for preservation. The tutor is Prof. Clemencia Chavez Lopez. Thank you.
Good morning, everyone. Today I am going to represent my research work, which is the cheese mold spoilage and strategies and new trends for the preservation. For the preservation.
You want to sit down?
No, no.
Okay.
Yes. The next one.
Oh.
I am working on a catchy cavallo cheese, which is one of the most important pasta filata cheese, which is commonly used in a southern part of the Italy. It is a pear-shaped cheese composed of the two spheres of the cheese, and the shape is given by hand, and the weight of the cheese is between from 3 to 5 kg, and the age of the cheese between 3 to 6 months. I am working on a cheese spillage, which is basically deteriorate the quality of the cheese, and in which the shelf life of the cheese is affected, and also the loss of the money, and the product become unnourished, and which lead to the dissatisfaction of the customers. And how does the spillage manifest? Particularly basing on the molds spillage, in which we see a visible growth of the colonies on the cheese food, and it also produce a gas, and also make our food slime, and it also produce some enzymes which lead to the production of the unpleasant smell. Different microorganisms are involved in a cheese spillage, but I am working only on the molds. For the control of the cheese spillage, different technologies and method are using, in which are the managing temperature, managing water activity, managing the presence of the oxygen, and also managing the formulation of the food, and accurate processing technologies. Nowadays, in a food, in industry, the antimicrobials are commonly used to control the shelf life of the cheese. There is a different sources of the antimicrobials which are using in a food safety. I am working on antimicrobials which sources are the plants. Here is a different type of the antimicrobials from the plant sources are used in cheese making, particularly for extension the shelf life of the cheese by controlling the growth of the unwanted microorganism. Basically, I am working in a project which title is process and product innovation in bovine cheese and a meat chain for human health, animal welfare and environment safeguard. During the period of my first year, first I work on a review of literature which is under a submission process, and the aims of my first year work is application of the bio preservatives in cheese production, and another is the effect of the antifungal compounds to increase the shelf life of the cheese in response to control the growth of the microorganism. Here, first I do the isolation of the filamentous fungi, which is responsible to cause cheese spoilage, particularly in a Caciocavallo cheese. Here we collect 6 samples of the Caciocavallo cheese from the same production house, and then these samples were analyzed for the isolation of the molds. Here you can see that we use a different culture media for the isolation and the purification of the cheese molds. First one is a cheese agar media, which we modified on the basis of which composition give a good growth to the molds. And after isolation, you can see that we isolate a different genre of the molds from the cheese, in which the large abundantly genre belonging to the Penicillium, and the another one belonging to the Fusarium and Alternaria, and the least one is Aspergillus. And we also observed a good growth of our molds on a modified cheese agar media. And then after isolation and identification of the molds, we will work on the control strategies, which basically focusing on the use of the essential oils, in which we use 5 different types of the essential oils to control the growth of the molds, in which we use a different concentration of the essential oils to check their antifungal effect on mycelial growth as well as on the spore germination inhibition, in which the three different type of the essential oil belonging to the Salvia rose marinus, the result shows that the reduction in mycelial growth mainly dependent on the concentration of the essential oil as well as the type of the molds, in which essential oil number one, the highest mycelial growth inhibition is observed on 7%, and the molds belonging to the Aspergillus genera, and another one in which we observed the reduction in mycelial growth of Aspergillus as well as Penicillium, and in another essential oil we observed the reduction in mycelial growth of the Alternaria. And these two essential oil are also used to check the antifungal effect on the mycelial growth of the molds, in which the Origanum vulgare at a 7% concentration give good control to the Alternaria, and in another case the Laurinus nobilis give good control to the Penicillium. Here we also check the antifungal effect of the essential oil on the spore germination. Here you can see that the all essential oils showed a spore inhibition result, particularly the essential oil number 5, Salvia rose marinus, give a good spore percent inhibition on Fusarium as well as Alternaria and Aspergillus. Ongoing work is that molecular identification of our isolated molds, and also I am currently working on a support time kill kinetics. I choose for this the essential oil number 5, which give good results, and as well as I am working on the antifungal effect of the polyethylene tetrafilate film, which is provided by the University of Bari, and currently I am working to check the antifungal effect of these 4 types of films which are coated with the essential oil or without essential oil. As well as I will also perform the analysis related to the physiochemical characteristics of the cacio cheese packed with a antifungal film, and also I will check the effect of these essential oils on the production of the mycotoxins, particularly citronine and ochra toxin, which these mycotoxins are really dangerous for our health. So that I will check the effect of the essential oils on the production of the mycotoxins which are produced by the different type of the molds, particularly fusarium. That's all. Thank you. Any question?
Thank you so much. Any question from the stage? Okay, I think we're done. Thank you.
Thank you.
So we ended this session and all the meeting. I'm particularly proud of these guys of the last session because they seem to be very in the deep of the project, and I think we all will be proud of them and of course of the other ones. I leave the floor to Professor Serafini for the last talks, and then we end the day.
Okay, quite easy. Thank you very much, everyone, also for staying here until the end. And just, now think about what you've done. How was your approach from the audience? If you are satisfied with what you presented, what your project, everything. Of course, this is an ongoing activity. This one was a little bit in a rush. So for next time, we have in mind different surprises. Let's say we try to also to interact with other PhD program in food. That would be improve interaction between us. So it's not just we just talk between us, but maybe interacting with other PhD student project would be extremely valuable. And we will identify some project and trying to PhD program and trying to make connection with them. For sure, there will be for next year, the day will be defined very in advance. So tutor and co-tutor don't have any kind of excuse to don't be here because we miss them, we want them. Considering for this year, we have many co-tutor and tutor. So they have to play with us and give us input. So thank you everyone. And if you have any feedback, anything, we are here to listen to you. Have a nice day.
Thank you.
Vámonos. Allora.
Infatti ti ho scritto, dicono
non lo so, è che devi fare una colpa, deve essere una cosa deficiente. Che ci devi fare? Non ci niente, non capivo, però